deployment of microbes in agri ecosystem. To initiate this process, gradually, I would invite my colleague, if we do start from our senior most faculty, Professor Ajit Verma on the dais. Professor Birinchi, please join us on the dais. Who is the keynote speaker today? Dr. Anukul, please come up. Dr. Amit Khadagwal, Deputy Director of AIMT, please join us. Please join us. I'm thinking to whom I should call to join on dais, but still we shall proceed henceforth. And without delay the process, I would request to all the dignitaries sitting on the dais to invite for lighting the lamp, which is parallelly will be invoked by my students, Miss Suniti Bhattacharjee, who shall deliver prayer of the goddess Saraswati henceforth. So Suniti and Monica, please assist us for incorporation of the lighting the lamp over here along with the Jagrati. Please.
Hello. हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। तू श्वेत वर्णी कमल पे विराजे, हाथों में वीणा मुकुट सर पे ताजे। तू श्वेत वर्णी कमल पे विराजे, हाथों में वीणा मुकुट सर पे ताजे। मन से हमारे मिटा के अंधेरे, हमको उजालों का संसार दे माँ। हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। तू स्वर की देवी, संगीत तुझसे, हर शब्द तेरा, हर गीत तुझसे। है हम अकेले, है हम अधूरे, तेरी शरण हमें प्यार दे माँ। हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। अज्ञानता से हमें तार दे माँ। अज्ञानता से हमें तार दे माँ। Thank you.
To start with the microbiome in climate change and food security. Before doing this, I would request my colleague, Deputy Director, Dr. Amit Khadagwal, please come up on the podium and provide glimpses about our institute. Please, Doctor.
A very warm good morning to one and all esteemed guests joined online and offline and dear students. So today I am going to provide a glimpse of AIMT under this SERB sponsored symposia on microbiome in climate change and food security held in MIT University. So we get our inspiration from our founder president, honorable founder president, sir, who want to make India's knowledge superpower by 2030. That is Amity's mission, Amity's passion, and Amity's junoon. And he famously says, when God pushes somebody, inspires somebody, leads somebody, no power in the world can stop such a person. And one such person he himself is, and we all aspire to become like him. And let me just give you introduction to AIMT. That is a DST FISH sponsored institute that is sponsored by DST, Funds for Infrastructure Support, that we got in the year 2016. I am Dr. Amit Kharkwal, Deputy Director and Associate Professor here. So AIMT was established way back in the year 2004 with 5 students enrolled in PhD and 6 in MSc at that time. And today we are offering various programs, BSc Honors Microbiology, our new latest program that is also now 8 years old, MSc Microbiology, PhD, and DSc in Microbial Technology. And if you see our student strength, we have 154 students in BSc and 88 students in postgraduate and 45 PhD students, and 2 ODC Viva are conducted this year. And pass out, if you see, we have 274 BSc, 318 MSc, 54 PhD. Our is the first institute to guide PhD student in MIT University and DSc 1 students, and 7 PhD degrees were awarded in the year 2022 itself. And AIMT is very well equipped with modern state-of-the-art facilities to do advanced research. As you can see from this list, all the major facilities are housed in AIMT. And there's some glimpses of modern microscopic facility, the confocal microscope provided by DST, the fermentation facility from BIRAC, big BIRAC projects, and atomic absorption spectrometer from Nanomission. All the instruments are sponsored from the sponsored projects. You can see nanoparticle size and zeta potential analyzer, gas chromatograph. This is our temperature, moisture, light controlled greenhouse. And very recently, Professor Dr. Ajit Verma, he has been awarded for a Lifetime Achievement Award in BioAg Asia Award 2022 by Agriculture Today Group by none other than former DG ICAR, Dr. Trilochan Mahapatra now. And if you see student achievement, student has won various awards and fellowships, DBT BIRAC, Sitare Geti Award, JRF NET, DST Inspire Fellowship, DART Fellowship, Marie Curie Fellowship, PhD scholarships in various countries in Western Europe, Australia, China, Scotland, 25, and postdoc fellowships in foreign countries too. So the main motto of MIT is we nurture talent. So how we nurture talent, that students are free to do research if they want to do research. All the students, you can get some guidance from a faculty guide, take guidance and start doing some research. This example of 2 young BSc students who did research, filed the patent, and as a consequence of that, got fellowship, 1 NOSI fellowship, right? And they are both doing very well now in their careers. Shubhangi Sharma, she got Marie Curie Early Stage Researcher Award, and now she has done PhD. When Prime Minister Modi visited last in Germany, Berlin, she was the part of delegate who met Prime Minister. You can see here. And Napreet Kaur is selected for Royal Bournemouth and Christchurch Hospital. She is working there. Khushbu Malhotra now has completed masters, and she has got job in England itself. She is working in London right now. Daisy Tyagi, she has also completed, now going for the PhD position in University of Halle, Germany. Srinivas Patil, he filed one patent with me and also presented paper in India International Science Festival and also contributed to book chapters. Sanskriti and Arpit Gupta, they published book chapters. Aditya Kumar filed one patent and now working in IRI. Gargi and Arpita published book chapters. Supriya, he filed one patent, now doing PhD in IT Delhi. Sneh Bajpayee in Thisti, she also filed one patent. Suryansh, he is in BHU, filed patent, published paper, and book. Preeti Sen Gupta also published book chapters. Vikrant Hari Kashyap, very brilliant student from AIMT. He got offers from 10 universities all over the world to join, PhD positions. And finally, he selected University of Sheffield, UK. So students, sky is the limit for you. Similarly, very brilliant student, Shubhangi Mahajan, who filed patents, published book chapters, books, and finally got, settled for PhD in Max Planck, Germany. Aditi Jain, she is now doing MBA in France in cosmetic management. And he co-edited one book. Chitrakshi published 2 book chapters. Ishan Tiwari, Ritika, they are still with us and they filed patent during their masters. Chesta and Manisha, they filed 2 patents, and the technology developed by them was selected by DWI, German House for Science and Innovation, New Delhi, as one of the top 15 innovations by young scientists from India. Rashmi Das, she filed 1 patent, still working with us. Madhunita and Purva Asrani, they got that fellowship for doing PhD in Germany. And Deepika Singh from 1720 batch, she got 2nd position in volleyball, so students excel in sports also. Ishan qualified CSIR UGC NET recently. Astha Rupali also qualified UGC NET, so students also qualify the exams. And faculty members, they go for the postdoctoral fellowships. So Dr. Ram Prasad, Dr. Neeraj, Dr. Swati, they're still with us. Dr. Swati Mohapatra, she did postdoc fellowship in Korea, and Dr. Kanchan Vishwakarma, right now she is in Sweden doing the postdoctoral fellowships. And this is the team AIMT led by Professor Dr. Ajit Verma, and eminent scientist, and young faculty members, as you can see, and you are all aware of about them. And this is the administrative staff of AIMT. So now we come to the business end, the research focus at MIT Institute of Microbial Technology. At AIMT, we are working in various domains like enzyme technology, microbial ecology, plant microbe and microbe interaction, plant genetic transformation, plant genome editing, nanosciences, environmental microbiology, and bioremediation. So students, you can pick all these areas if you want to do some research. And the faculty expertise is listed here. You can take a pick of this, and according to the area of your interest, you can meet the the faculty members: plant microbe interaction, enzyme technology, plant genome editing, molecular biology, plant genetic transformation, microbial ecology, environmental microbiology, so bioremediation and nanoscience. So there are many areas for you to choose. And faculty have won various awards and fellowship in last 20 years or so, and some of these are listed here. And faculty also comes in world ranking of top scientists, Professor Dr. Ajit Verma in world ranking of top 2% scientists, and Dr. D.K. Chaudhary in the list of Indian researchers who were in top 2% Stanford study. And if you see publications and patents, 368 publications in last 5 years with average impact factor of 3.5, book chapters 215, edited books 70, and total books published from 120 is 128 from AIMT. That is a sort of world record. We have filed 53 patents and 10 patents are granted to us. And ongoing research projects worth 88.63 lakhs we are doing right now. And when we say patent, technology, we also develop technology, transfer it to industry for Atmanirbhar Bharat. That is novel innovations to make India self-reliant. That is the vision of our Prime Minister also. And we have, towards that, we have developed novel innovative technologies for sustainable and climate resilient agriculture. So that is the theme of this symposium today also. So this is the technology of Rutonic. So memoirs of Rutonic, you can easily download it from net, right? And we have developed this fungus. Professor Dr. Ajit Verma has discovered this fungus, and this you can see the world's first culturable mycorrhiza. Mycorrhiza, all of you know that they are non-culturable in the synthetic media. So this is the world's first culturable mycorrhiza fungus, and it can be grown on a semi-synthetic medium, as you can see. And when it is applied to seeds, seedlings, or standing crops, it provides tolerance to biotic and abiotic stress, and also increases the yield of the crops. So here you can see this bottle guard field control and treated not only disease suppression but yield enhancement was also there. Up close you can see how this fungus P. indica, that is root tonic, is suppressing the disease resistance to pathogenic fungus. One more important thing about this fungus was discovered by Professor Dr. Ajit Verma from the western Thar deserts in Rajasthan. And this fungus was tested in the extreme cold condition of Leh Ladakh, and there also you can see it promoted plant growth and early germination and early development of plant under extreme cold condition. Recently, we have isolated one more strain of species Teloromyces, as you can see. So this is also plant root colonizing endophytic fungus, as you can see in this picture, and this having various plant growth promoting traits like zinc, phosphate, and iron solubilization, indole acetic acid production. And this is how the interaction took place in greenhouse. So increase in the plant growth and root growth, root architecture, as you can see. And this is a root colonizing fungus. And we interacted here in the field also. And you can see early flowering, more, bigger stand size, and early fruiting took place. And naturally, recently we have done one more innovation with the culture filtrate of this fungus, that we are able to make a natural fabric. You can see here control wool and the culture filtrate treated wool. So culture filtrate can be used as a natural dye and as a natural color also. Because it is a non-mycotoxin producing fungi, so you can use it for natural colors and natural dye for food and textile industry. And one more innovation that is under commercialization, that P. indica prolongs the shelf life of orchid cut flower. This you can see the cut flowers on day 0 and day 35. You see the orchid cut flowers are still fresh. So this is the magic of the technology. And we have transferred the technology to various companies, Sanan Inc., Sai Bioorganics, Som Phytopharma, Sri Bioaesthetics, and recently to one more company we have done, Vedic Vigyan Kendra, Ayusham, for HNB9, and MOU and material transfer agreement with Premier Tech, AMC Chemicals. ICL Innovation has also sponsored our research recently. They have given us financial assistance, also in addition to signing the MOU for material transfer. And negotiations are under progress with many companies like Ariona, Biotech, Hongxing, and Plant Promotion, and many other companies. So this is the new product in the market, Kriya DMF. Kriya means action, a Sanskrit word that is action. Of DMF means desert mycorrhiza fungus, that is Piriformospora indica. And these are the books which were published in 2020, 2021, and books of 2022. So this is the activity calendar of 2022-23, and you can see we have developed faculty development program, with Indian and German scientists and members from industry, inauguration of new established practical laboratory where you are working in G Block, and SERB DST sponsored workshop of advanced microscopy was conducted at MIT campus for sustainable agriculture. This is the picture with the participants. And then DBT sponsored international conference on emerging trends in genome editing was also conducted on 17-18 August. This is the participants, with us. And so thank you very much. All the best. Thank you once again.
Surely, we are very thankful to Dr. Amit Khadagwal, Deputy Director of AIMT, who provided nice glimpses. And the foremost things comes in mind, how diverse is diverse. So you may see that we deployed with the diversity in the students, diversity in the faculty colleagues, and diversity in the microbes as well, you know. So bacteria to which one? Which domain we should discuss with all these scenario nowadays? Gracilicutes, firmicutes, molecules, mandosecutes, and their incorporations with the soil and rhizospheric microflora as well. So we shall dig out later. So with this, I would request respected Professor Ajit Verma for opening remark. Sure. Dr. Anu, please provide help for sir. So you may reach us quickly. Who is online? Dr. H.P. Singh. We welcome Dr. H.B. Singh.
Online, sir. Just traveling, but I'm online.
Yeah. You told me that some investment meet taking place at
Yeah,
Lucknow.
yeah. But
So-
maybe around 1:30 or 1:45, I'll be.
Okay, okay. Our students will be benefited by your talk at 1:50 around, or quarter to 2 p.m.
Yeah.
Please.
Thank you. Good morning, everybody. We welcome the guests from outside, our dear colleagues, and my dear students. What a great occasion today to hold this symposium. I'm very pleased to inform and remind you that in the last 1 year, we have conducted several such programs supported by DST and DBT. It is a rare honor for any institute to get so many commitments from DST and DBT. There was one held on the faculty development program, and it was held in April 2022. Then 6th International Fascination Plants Day on 19th May 2022. Then workshop on advanced microscopy for sustainable agriculture. This was held in July 2022. Next was colloquium on emerging trends in genome editing. It was held in 17th, 18th August 2022. Next was 18th AIMT Foundation Day. It was held on 23rd September. It should be 24th, but this happened to be a holiday, so we preponed to 23rd of September. The workshop on advances in microscopy in biological system was held in 17th to 19th October. So this is a long list of program we held in AIMT. This reflects that our institute, although small, but is very active with academic activities. Our students are very bright. They are asked to start research right when they join the MIT University as a BSc honors student. They continue for MSc, and by the time they come to the BSc, complete MSc, they get publications, they get patents, and they also get books. This helps them to get secure good position in any university all over the world. Precisely, our students are being accepted all over the world, and also job in India. They are bright, they are very bright. We are very proud of them and very enthusiastic, working hard till late in the evening. Conduct practical. One thing I like to tell you that in our institute, all practicals are conducted in reality. The students are asked to use the instrument and learn this capability. I know I come from Allahabad University, limited resources, limited instruments, and we are not allowed to use the instrument, they will get damaged. When I went to Australia in 1967, I was suffering an inferiority complex that I don't know how to use the instrument. Learning demonstration from distance is different than what we actually use. Since then, I had decided that all the time, students must use the instrument themselves. Let them get spoiled, doesn't matter, we'll repair it. So when I joined JNU, this was a momentum in the microbiology center. All instruments must use all the instruments. They will learn a lot. By operating the instrument, you will learn a lot. Small pitfalls. I'd like to add what Amit rightly pointed out. We've got many patents. One more patent was filed, and it came for minor repairs, changes last week. It was done. So we are very proud of our patents, and most important, commercializing them. We have commercialized these patents, and by now we have earned almost 1 crore rupees from the patents. And we are pursuing many patents to be commercialized. This is the important point. And this information is passed on to the student. Let them also cause patents. Let them also file a publication patent. Is Sikha Solanki here? Just please tell me that I'm not happy for her absence. All PhD scholars must be here. Okay, good. So, but you say something? Okay, I thought you were going to say something. Okay, good. So I hope by now you got enough information from Dr. Amit Khargwal, my colleague, very competent colleague, and our most competent colleague, Dr. D.K. Chaudhary, has organized it. I'm very thankful to him for steering this program very nicely. And one thing more, that he is going to have another program next month, which we were talking last week. He got grant from DBT for another program. This reflects the recognition of our university, right? Oh, welcome, sir. Welcome, welcome. Dr. Selamurthy, yes. Now I have finished, you can continue. It was my honor to introduce you Dr. Selva Murthy, W. Selva Murthy, the President ASTF. He has kindly consented to come. He was held up in traffic. But look at his efficiency. He called me, I'm held up, I'll be held up by another half an hour, but I'll take the program online. And I should be here by 10:30. Straight, I'll come to this hall. That shows his commitment to the science program. He's our guru in science matters. We always consult him for everything, small or big. Even we do mistake, we consult him and tell him. And he's very kind to us. Thank you, sir. Thank you all. So I request Dr. Selamurthy to come and address the students. Thank you, sir. You go ahead. Yeah. Yeah, very kind. Yeah,
My dear students, distinguished faculties, delegates, resource speakers, invitees, on behalf of Founder President and on behalf of the Chancellor and on behalf of everyone from Amity Fraternity, it gives me immense pleasure to welcome all the participants at this very important SERB-sponsored symposium on microbiome in climate change and food security. So Honorable Founder President has attached a lot of importance for this symposium, and he will also be listening to all the deliberations and So he would be also maybe giving his blessings at the end of the deliberations today and tomorrow. Let me particularly welcome our guests who are on the dais, B.K. Sharma from BHU and Dr. Anukul Vaishnai from GLA University, who is also passed out from the portals of this university, particularly under the guidance of Dr. D.K. Chaudhary, and all the very distinguished people who will be joining, Dr. H.P. Singh, Dr. Amitav from BHU. Then we will also have Vishal Tripathi, Dr. Praful, Dr. Vaibhav Srivastava, Dr. Shekhar Jain, Dr. Siddharth Tiwari, Dr. Anil K. Sharma, and many more distinguished people who have joined online and will be giving their address, sharing their expertise, knowledge, wisdom with all of you. So let me welcome all of you for this very important symposium. Today, if you really look at India, it's a new, vibrant India. So particularly the youngsters, the young India, where the average age of an Indian is about 27, 28. So this is the youngest country today in the world. So that is why the whole world is looking towards India because of its rich heritage. We had agricultural era in which we were teaching the whole world how to cultivate agriculture, how to cultivate crops. So India has a rich heritage, rich civilization, and rich knowledge. Vedas, Upanishad, Bhagavad Gita, Yoga, Ayurveda, all this existed in our country. It was a knowledge superpower. So now the world is looking how India will emerge as a new knowledge power again, and also new global power, maybe superpower. So that is why this symposium is very important, because the symposium is being held at this very, very important transition, Amrit Kal, which our Honorable Prime Minister calls it as Amrit Kal. When you look at 75 years of independence, Azadi Ka Amrit Mahotsav, which we are celebrating, India was a begging bowl at the time of independence. We had 30 crore population. Now it is more than 140 crores. 30 crores, we were not able to feed. The food has to come from another country through PL 480. But today, that country, which was a begging bowl, is a bread basket today. We are able to export food. We are able to give a food to another country in calamities like tsunami, earthquake. Whenever there is a need, we are able to give assistance, aid, food aid to the people besides meeting our requirement. So that transition occurred because of scientists, application of science, technology, with participation of farmers, political commitment, bureaucratic governance, supporting the Green Revolution. Professor M.S. Swaminathan, working with International Rice Research Institute and other organizations, we were able to bring the transformation. But today, we are also faced by many challenges. That was one challenge which we faced effectively, but the new India is going to face many challenges in this field. The first thing is we have to feed 140 crores. That was 30 crore, 140 crores, which is still growing. And India is now almost the most populous country in the world. Even we are overtaking China. So if that is so, those people have to be fed. Now the middle class, the purchase parity, when you look at that, the purchase power has increased, consumerism. We are consuming more food. So the demand is rising, not only due to population, but due to purchase parity. The second aspect is the cultivable land today is dwindling down. The soil health is eroding. We are building structures, factories, the living apartments. All this is being built on agricultural land. So now the cultivable land is dwindling down in terms of size. The other is soil health is eroding because of over exploitation of the use of fertilizers, pesticides. We have eroded the health of the soil, salinity of the soil, alkalinity of the soil. And all this is now another change is soil health erosion. The other important issue is the climate change, which is looming large at us. They are looking at us. How are we going to handle this climate change? And that is going to affect the biosphere, including our cropping, agriculture. So you see the challenges for all of us, but then human mind potential is infinite. Any problem, we'll find more than one solution. That is Indian intellect, as well as the power of human mind. So we will be able to tackle this with our resilience, with our intellect, with our application, with our dedication. India will find solution. That is why the new India in agriculture and food security and climate change, we are going to face these challenges. So children who are all in this field, life sciences, agriculture sciences, you just get ready, awake, arise, and stop not till the goal is reached, Vivekanandaji. So let us awake ourselves. Let us awake our Kundalini Shakti. Let us arise from our the normal comfort zone if people are in comfort zone, and then let us not stop, because the life is very short and miles to go before we sleep. So that is why let us just make a determination, young students, faculties, and all of us on the dais, online. Let us take a resolution. In the new India, Amrit Kaal, 25 years, we are going to rise exponentially, leaps and bounds, pole vaulting in science and technology. All this will happen. So let us prepare for this new India. And we are preparing you students to face those challenges. So you understand the responsibilities on your head. Let's clap for our brilliant students. You are going to face those challenges. If you look at the centuries, let's start from 16th century. We had agricultural era. In agricultural era, India did contribute significantly. But when it came to industrial era, because of the British colonial rule, India did not participate, could not exploit that opportunity of the industrial revolution. But then information era came between 1960 to 2000, where India played a very important role to utilize the information technology revolution in service sector and earned a lot of revenue. And the 60% GDP of India came from service sector using IT revolution. But in this new knowledge era, India is going to play a very important role. And we are all going to contribute a significant role in transforming, creating a new world, which is seen by good health, food for everyone. Everyone, no one is dying of hunger. So we are going to create, India is going to create a new world, which will be good health, happiness, peace, prosperity for everyone. So that is the world, that is the mission in which Dr. Ashok Chauhan, the founder president, is creating, preparing the future global citizens who are sitting here in the dais, who are in all our 11 universities in India, and the 18 campuses abroad, 28 schools, 2 lakh students, 10,000 faculties. We are all in that mission of Dr. Ashok Chauhan to create that new world, preparing our future global citizens. So this is why your role, our role is very, very important. If you look at the 19th century, it was occupied, prominence was there in physical sciences, a lot of physical science, Newton's law, Einstein's theory of relativity. And so like that, there are many, many science, the physical sciences was getting a focus. But when you came to the next 20th century, you will find importance goes to engineering as well as technology, industrial revolution. Then came the 21st century that belongs to life sciences. That belongs to you, all these people who are sitting here. 21st century belongs to life sciences. That is where the microbiome, is going to play a very significant role, the microbiome, soil microbiome, plant microbiome, gut microbiome. So when you say microbiome in the biosphere, we only see the animals, we see the human beings, we see the plants, algae, but there are microbiome which is there, which is occupying a much more space in the biosphere, which we don't understand yet. So that is why microbiome, which is the theme of this, is very important. This microbiome, which we are going to talk about, related to agriculture, plant, food, so which is going to be very important. So all of you have a tremendous opportunity to explore this new venture. Amity University, which is focusing in this area, Dr. I was listening while I was coming in this right from the beginning, right from the light lamping. Amit Karkwal, you did a great job to project all the achievements of Amity Institute of Microbial Technology. This is one of the premier institute which Dr. Ashok Chauhan and Dr. Ajit Verma established way ahead. In 2004 itself, before it became university, Dr. Ajit Verma, who was an expert in JNU, was brought to Amity because Dr. Ashok Chauhan, founder president, keeps looking for talents. So he was one of the talents who was brought early when the university was established here. So this was one of the premier institutes. So when Amit Karkwal was presenting the students' achievements, where our students are doing, alumni are in great positions, great laboratories, working with great people across the globe. So it gives satisfaction to all our faculties who have groomed them like this. And under the leadership of Professor Ajit Verma, Dr. Amit Karkwal, Dr. now D.K. Chowdhury, and all those distinguished faculties who are sitting here. So these people have groomed. So I was so happy to listen that the students are doing well and the faculties are doing extremely well in terms of the number of publications, patents, the technology transfer. This is one institute which has done more than 5 technology transfers, which is using the Piriforma spora indica, which we call as root tonics. For root, it's a tonic. It's a root endophyte, Piriforma spora indica, which gives protection against the abiotic stress, low water availability, high temperature, low temperature, salinity of the soil, alkalinity of the soil. Against all this abiotic stress, this microbe is a magic microbe. He calls it as magic fungus. Dr. Ajit Verma, who discovered this in the deserts of Rajasthan, which works also in cold desert in Ladakh. So he found this and he has transferred this technology to 5 industries. We have PCT, global protection. We have patents. I think 8 patents are there only in this microbe in different processes. Now they are looking at a microbial consortium, not just one fungus, but then also like Acetobacter. And can we make a consortium of microbes which can do much better than a single microbe, which is Piriforma spora. So he is looking at the synergistic, the microbes which can become a consortium and which can meet climate resilience, because climate change requires a different type of challenge. So how do we take care of the climate change using the consortium of microbes is another focus in this. Then Amity University, in terms of research, I should tell you that with all humility, under the able guidance, drive, and the leadership of Dr. Ashok Chauhan, we have been able to file 1,870 patents. Clap, let's clap. 1,870 patents. It is for a young university, which is just 17 years young. We became university in 2005. This is a great number. And we were awarded by Government of India, Ministry of Commerce and Industries, Mr. Piyush Goyal, the Honorable Minister. He presented, and Dr. Ashok Chauhan, the founder president, received. On 17th August 2021, this award was granted for top academic institution, for patents and commercialization. It's just not only filing patents, but we also commercialize. We translated them into product, technology, process, and then transferred to industry to reach out to the people. So this is Amity University. And we have published more than 30,000 research papers in journals of repute like Nature, Lancet, New England Journal of Medicine, the Cell, Royal Society of Chemistry journals, IEEE journals, and advanced materials, Material Horizon. So our H indexes, now we have reached 110. Our H index of Amity University, the whole university, is 110. But then that's not good enough. So we are now looking at, Chancellor has given a goal that we should reach 140 within another 6 months to 8 months. So we are working towards that. So I should also appreciate the Amity Institute of this microbial technology, which is doing a great job, which you have seen. And there are Dr. Ajit Verma, Dr. D.K. Chowdhury, all of them are in top 2% scientists in that Stanford survey, which has been done. We must also record our appreciation and gratitudes to DST, who has sponsored this symposium, the Science Engineering Research Board of the Ministry of Science and Technology. They are always supporting. I want to tell you that Amity is one of the largest university which has major projects from DST, Ministry of Science and Technology, DST, DBT, fellowships, all this, whether it is SIRE fellowship or it is the Ramalinga Swami Fellows, Ramanujan Fellows. We have 40 of these distinguished postdoctoral fellows here in Amity. Ramalinga Swami Fellows, Ramanujan Fellows, Inspire Faculties, Welcome Trust Fellows, and also National Postdoctoral Fellows. So your university, Amity University, is bestowed with that honor and support from DST. Let's clap for all officials of DST, SERB, because our country today, when you look at the world merit list, in scientific publications, India is number 3 in the world in terms of number of the research publications in journals and other publications. When you look at patents, we are 9 in the world. We are number 9 in the world. And when we look at the number of PhDs, we are number 3 in the world. And so where we are now lagging is Global Innovation Index, Global Innovation Index. So now our next step is, and founder president has already set a goal, we should be in top 10. Why top 20, 25? From 40 where we are, we moved to 40 from 81. That's a big change. That's a big growth. But then we are not satisfied with a position of 40 for a country like ours with all the intellectuals here. So that is why it is our goal. Soon we should come to the top 10 position in which Amity will contribute a big way. So we have given a focus in this year onwards on translational research. So we have established Amity Institute of Translational Research. Dr. Hridesh, you just came at the right time. Hridesh is now looking after this, nurturing, but then many more will join, many faculties, all of you have to join this effort, Amity Center for Translational Research. So the translation means you are not stopping with publication, patents, publications, but we are translating those leads into a deliverable, a product, technology, process. So that will meet the Atmanirbhar Bharat, which our Ajit Verma Sahab also mentioned in his speech. And also Amit Karakwal presented in the slide, Honorable Prime Minister's vision. He drives with slogans and visions and the intrinsic motivation of this 1.4 billion people. So his main aim is Atmanirbhar Bharat. We should be self-reliant in everything, every technology, defense, agriculture, food, energy, everywhere. But when we are doing all this, India is also conscious of sustainability, sustainable development goals of United Nations. So we want to have sustainable agriculture. That is where microbiome comes. We want to get rid of pesticides, fertilizers, biofertilizer, biopesticide. So this symposium is talking about microbiome, which will replace or reduce the use of fertilizers, pesticides. So that is why this symposium is very, very important. So I'm so happy. And founder president also has conveyed his blessings to all of you. Now, the final message I want to give India will become carbon neutral by 2070. This is again a commitment country has made. Honorable Prime Minister has made on our behalf. So if you have to become carbon neutral by 2070, which is not very far, maybe another 45, 50 years, for which microbiome application is one of the important steps, because this will reduce the carbon signature in agriculture. So that is why let us give a focus on this translational part. With this, I wish you all the best for the deliberations. But founder president always mentioned many missions, but one of the two missions is very interesting, very important. One is mission synergy. We are all working in silos. We are islands of excellence. How do we transform ourselves into continent of excellence, connecting with each other, working in synergy, networking with each other, synergizing the efforts of the other team, other institution, other organization. That is why we have signed an MOU with many organizations, ICAR, Indian Council of Agriculture Research. We have a formal MOU, Amity Group can seamlessly work with all ICAR institutions, ICMR institutions, DRDO institutes, CSIR institutes. So this synergy is very, very important. So one mission is mission synergy. The second mission, which I want to convey here for this symposium, is mission outcome. We deliberate, we bring proceedings, we organize symposiums, seminars, webinars, workshops. But what is the outcome? Sustainable outcome, not just outcome, sustainable outcome. So we would expect from this symposium also, SERB sponsored symposium, the sustainable outcome. So we would be eagerly looking forward, Dr. Ajit Verma, Dr. D.K. Chaudhary, Dr. Amit Karkwal. We'll be waiting for your outcome report and follow up by our Directorate of Outcome from Group Captain Kapil Shukla. And then how would you take it forward? What are the specific, concrete, implementable recommendations you will provide from this symposium? So we will be eagerly looking forward to those outcomes. And with this, once again, thank you very much for this opportunity to share my thoughts. And so we, once again, blessings from Honorable Founder President as well. Thank you so much.
Certainly, we have blessings from all the dignitaries who are on the dais, and Honorable Founder President has also blessed his showering on us. So without, based on the defined program schedule herein, I would request to all of you, please join us for networking tea located outside. And thereupon we shall start with the how do we conceive this idea to pursue henceforth and convene this deployment of the microbiome in sustainable agriculture based on the climatic impact on the plant holobiont. So I would request to all of you, my colleague Dr. HP is here with us also. So please join us for tea, and we shall gather thereupon after half an hour. Please join us. We must move ahead. Upon persuasion of high tea, we all got energy and allow microorganisms to do their work. Gut microbiome. So why do we conceive this idea? It was a perception in the mind that we all are working in same area for last many years. And Professor Ajit Verma, a well-known figure globally, we are following him. He deals with the fungus. His associates deal with fungus. And one of us dealing with the bacterial system as well in the plant microbe interactions. So what is the utility of that microorganisms? What are they doing in the soil, in the rhizosphere? and wholly, I would say the plant holobiont. In recent time, my team, including Dr. Anukul, Dr. Shekhar, and others also, edited a volume for Frontiers in Microbiology. It relates with the climatic impact on plant holobiont, mitigation strategies, and sustainability. We wrote an editorial for this journal, which defines that under what situations, we can deploy microbes to alleviate habitat imposed stresses, which may be caused by the biotic factors and abiotic factors, especially the salinity and drought, if we talk about abiotic stress, and many phytopathogens that may be included from the fungal diversity as well. Somehow, we are missing microfauna in rhizosphere. We always try to capture the diversity of the microflora in alleviation of the stresses, whereby we may ameliorate, we may fertile the soil, and permitting the crop productivity as well. Few years back, we made contact to the German scientist, Bolonsky, who is working on protozoans. Rhizosphere diversity of protozoans. Novel things came in our mind, and we started to find out what we do for the exploration of the protozoans, because we are wandering to find out the microflora in the domain bacteria, and still we are lagging behind to capture the archaeal diversity also in the rhizosphere and soil. What do they do? What are their importance? And you all are young buddies going to be a scientist later on upon pervasiveness of the scientific scenario. So try to explore these area in coming years. Protozoans diversity in the soil and rhizosphere, and archaeal diversity. So beyond, so you shall try to achieve all these three domains: bacteria, archaea, and eukarya overall. Now, we were thinking a few months back with the chair, Professor Ajit Verma sir, we must move in exploration of the microorganisms to organize some kind of the functional activities that may be incurred as a plant holobiont. We always try to talk about the epiphytic microflora, endophytic microflora, and their interactions with the plant as a crosstalk. Being a microbiologist, we are always trying to dig out the four classes: the gracilicutes, that defines the gram-negative bacteria, firmicutes, gram-positive bacteria, molecules, mycoplasma-like organisms, and mandosicutes like archaea. As name suggests, if you see this theme, microbiome in climate change and food security. Climate change, what does it mean, climate change? It may be invoked by the action of microbes in emission of the gases, global warming, climatic impact also, nitrous oxide, methane, carbon dioxide, and so forth. So how you are going to alleviate such stresses in the system, whereby you shall provide the amicable environment for the soil and the crop to cultivate in a feasible fashion. So interlinking with these aspects, we designed this symposia, especially by keeping the view of Sustainable Development Goals. Number 2, no hunger must be there globally. Along with the SDG 1, no poverty must be laid upon. So how do we are going to intricate the microbial diversity to alleviate or to achieve sustainable development goals 2 and 13 as well. 13 defines for the climate impact. Recently, we edited a volume under guidance of Professor Verma, the climate impact and sustenance of ecosphere, wherein we emphasized the role of the microbial diversity in alleviation or diminishing such gas emissions also. Because if you are working in archaeal diversity, the methanogens always comes into the force. So methane secretion and its pooling in the biosphere, which may causing the damaging impact on the ecosystem also. Likewise, CO2. So how do microbes do in capturing of these gases? In what way? All right. Do they adsorb? Do they absorb these gases? And if we talk about the methylotrophs, they use methane or the methyl compounds as a carbon sources as well. CO2 definitely photosynthesis, if you remember. So we are going to capture the CO2, but it is not beyond the limit. The presence of the light is necessary for the fixation of the CO2, which is incurred upon action of the Rubisco, you know. So it is the largest enzyme or the functional proteins on the biosphere that has been reported. There are several important points I must disclose to all of you by keeping the few words provided through Dr. M.S. Swaminathan, sir. And everyone knows about M.S. Swaminathan. He always tried to say that nowadays the molecular or modern tools must be there to explore the microbial diversity in an ecosystem. So according to him, this is not my word, but these words belong to Swaminathan, sir. Access to information through ICT or any other approaches that will equip the farmer with necessary tools to fight low productivity or even the effects of climatic impact. And again, he includes few important lines that we need fund to pursue research to alleviate such kind of the strategies, mitigation of the climatic impact, and the involvement of the microbiome as a whole. So while expressing the concern over the lack of funding for research, Swaminathan sir voiced an optimistic note that it is true that agricultural productivity in India is very low. Not a low in that way, but as compared to the US, China, and Brazil, somewhere we are lagging behind. So we lag behind with many countries. However, this is also a cause for optimism since we can improve our productivity. So we can ensure that issues related to food security can be fixed and we can fight climate change while ensuring no stomach in the country goes hungry. So it implies 2 SDG 2. So number of approaches, the plant geneticists, plant breeders, they are plant biotechnologists also, they are deploying their way of explorations, how to survive the plant upon implications of the climatic changes. And we, today, we are deploying a biological agents for agriculture. So it may be recovered by the action of the endophyte. Like P. indica, well known, well renowned, well published impacts of the P. indica for last many years under Professor Ajit Verma, sir. And thereupon, we also trying to contribute through the bacterial system as well. And Professor B.K. Sharma, a keynote speaker for today's talk, he shall provide glimpses on the microbiome aspects in agricultural productivity, and which will be followed by my colleague, Dr. Anukul, later on. I would like to quote a few important points over here. CIMIT Institute located in Mexico, right? The director or CEO, the Arun Joshi, which is looking considering this representative from Indian scenario, working at the International Maize and Wheat Improvement Center. According to him, the climate change mainly emphasized on the rising of the temperature. He emphasized temperature over there. So 1 degree Celsius rise in temperature impact the whole crop productivity henceforth. Beside that, I would like to quote one important thing from MS Swaminathan Research Foundation, Madhura Swaminathan, which concurs on the need for a diverse diet also, which indicates fruit and vegetables in terms of horticulture, but points out that the fight against climate change from a nutritional perspective will be different in the western part or in the US and UK countries along with in India. So we must not forget the deployment of the microorganisms in form of nutraceuticals also, which elicitors or which determinants the microorganisms playing important role. So if we talk about the determinants, so we all are working somehow in the explorations of the volatile organic compounds, 1-aminocyclopropane carboxylate deaminase activity, relativity of the compatible solutes produced by the microorganisms and their implications further in the plant system, whereby the plant you may cultivate from the fertile land to the arid land or the semi-arid land. One more institute in India, ICRISAT, we are much familiar with the ICRISAT also in Andhra Pradesh. So ICRISAT peoples are working in that way to produce, and they are mostly revolving around the leguminous plants cultivations in the semi-arid tropics. So they use chickpea, and upon occurrence of and modifications by helping of the microorganisms and under the cultivations, they provided a note that chickpea has the ability to provide more nutrition and are resistant to the climate change. So if you want to cultivate a high altitude plant, which is growing in a normal fashions in an area which is arid and semi-arid, so what kind of the changes you are going to incorporate in that plant? Genetics, recombination aspects, mutational aspects, and above all, why we shouldn't work on the role of the microbiome, which may help in sustenance of that plants under such adverse environment. Yesterday, a note was there. I was watching a DD News channel. So they were talking about the deployment of the biofertilizer nowadays in India to enhance the agricultural productivity. Dr. Malhotra was there on the channel. He was talking about the deployment of biofertilizer and biopesticides. Why we are not deploying this? Still, farmers are not familiar with the concept. So it is a translational approach that we shall convey to them. With this, we made request to Professor Manju Sharma, Madam, also to write a message for us so we can convey her message to the society. And Dr. Manju Sharma, presently, she is a chairman, New Initiative, NASI, Allahabad. So being a member of the NASI, we made request to her, Madam, please provide few lines which may be conveyed to the young minds and the society about this talk. She wrote, I am happy to note that the present symposia is being organized at a very relevant time, because we are facing drought conditions, salinity conditions, and other biotic and abiotic factors. According to her, I congratulate the organizers, we all of us, for the same. And the subject of the present symposia is very important and relevant for the socio-economic progresses of the nation, particularly considering the critical aspect of the climate change and its impact on economy, and also a very burning issue of food security to combat malnutrition. Madam said that I am sure the participants will be the students. Yes, definitely, Madam. We have more than 200 students with us, young minds, and the young scientists interested in pursuing a research career to contribute toward research, innovation, technology, and finally outreach programs. So what will be the outcome of this symposia? We shall make a report to her also. Microbiome R&D is important from the viewpoint of food, feed, and other therapeutic aspects. We always try to make talk about the therapeutic dose, usage of dose in a therapeutic way. So what will be the actual threshold or optimistic value of that dose that we are dealing with the plant system? The same way we always try to imply towards the human system also. We always try to take a threshold or optimistic level of any drug or the pill. So more question comes around the low affinity and high affinity of the receptors located on the cytoplasmic membrane. If dose is not effective, then we take high dose to be fixed on the target for the further cellular and molecular interactions through the signaling processes. So dose dependent interaction is necessary, and it is a matter of the considerations of the target or the receptors. What do they do for intake of actions? So all the area having significant advances have proved that they are not only important for the human physiology and benefits of the health, but also contribute toward the environmental safety and food security. Need for taxonomist. She is emphasizing the role of taxonomist also, because at the start, at the onset of this convene, we talked about the gracillicutes, firmicutes, molecutes, and the mandosicutes. So what do they do? So this is a deployment of all these microbial or the bacterial classes along with the fungal phylums over there. Asco, Basidio, Deutero, and Zygomycetes, and henceforth. So everyone knows about the microbial phenomenon. Being a microbiology students, you are much familiar as we are dealing with you on everyday aspects. So Madam said that it is a need for taxonomists that has been recognized widely across the world. The microbiology as a subject is gaining tremendous significance. So we really need multidisciplinary team in a holistic way to work in these areas so that the results may be applicable for the human welfare, safety of the environment, and preventions from the natural calamities. I wish the symposium great success and my best wishes for the students and the scientists who will benefit it from this. Manju Sharma, Madam. So we got this message from Madam. Another important message we received from Dr. Himanshu Pathak, who is the Secretary, Department of Agricultural Research and Education and Indian Council of Agricultural Research, ICAR. So these two personalities provided a message. So I defined a message laid upon by respected Dr. Manju Sharma, Madam. Here, based on Himanshu Pathak, sir, he wrote 2 to 3 para in a gist way. Please listen carefully. According to him, I am pleased to learn that Amity University, Noida is organizing SERB sponsored symposium on microbiome in climate change and food security. So global food demand is increasing and the available reveal that we will have to produce about 70% more food to feed the world's populations by 2050. So it comes under the SDGs defined by the United Nations. 17 SDGs we must fulfill by deploying this SDG 1, 2, 11, 13, and 15 over here. Sustainability, ecosystem sustainability, alleviations of the climate change, and so forth. So being a scientific minds that you all are having over here, we have to focus on these aspects. So I would read one more para written by him. I hope this symposium shall provide adequate opportunity for serious deliberations to scientists, scholars, students, and scientific fraternity to develop and implement the effective strategy for the implementations of climate mitigation and mitigation policies to facilitate the governance and management of the climate changes. Besides, symposia will help to contribute and bring leading academicians and scientists together to exchange and share their experiences about all aspects of microbiome research. I wish the symposium is a great success. Himanshu Pathak. So with this, I would welcome our keynote speaker, Dr. Birinchi Sharma. Sir, please enlighten us with the your glimpses that you have incorporated in your presentations. So students, please welcome Dr. B.K. Sharma, sir.
A very warm good afternoon to everyone present here. Now, we would like to invite Dr. Birinchi Kumar Sarma for his talk on microbiome, potential to mitigate soil-borne diseases and salinity stress in crop plants. He is a professor of plant pathology in the Department of Mycology and Plant Pathology, Banaras Hindu University, BHU. He obtained PhD from BHU in 2002 and was a postdoctoral researcher at University of California, USA. His research focuses on legume pathogen interactions in saline soils with emphasis on understanding transcriptional regulation of fusarium wilt susceptibility in legumes in salinity-stressed soil, legume genomics, and biological control of legume diseases. He has filed 5 patents, edited 5 books, and published over 80 scientific papers and review articles. He is a fellow of Biotech Research Society, India, associate of NAS, editor of BMC Microbiology and PLOS ONE, and recipient of Boy Scout Fellowship of Department of Science and Technology, New Delhi. A very warm welcome to you, sir.
So very good afternoon to all of you. It's a great pleasure for me to be here, especially meeting Professor Ajit Verma sir, who has inspired a lot, including me. We have read his articles about P. indica in great depth and generated a lot of ideas from his work. So it's thank you, Dr. Chaudhary, for giving me this opportunity not to address this gathering, but to meet sir as well. So as the topic I selected, that is microbiome as a potential to mitigate soil-borne diseases and salinity stress in crop plants, I will try to address some of the work that we are doing in our laboratory, and in general, how microbiome, the area is flourishing, and to understand different aspects of soil and climate factors and how to mitigate them. I will also take up few examples from other labs who has published some very important data to understand the soil microbiome in such in totality. So coming to the topic, Dr. Chaudhary has already mentioned that we have certain SDGs, and number 2 is zero hunger. So being in the field of agriculture, we all are working on that particular aspect, and for attaining zero hunger, we need to actually secure food. Now, this is a very common term, that food security, but how do we actually understand this term and why we should really care about it? So that is my first observation, and especially the students are sitting over here. So I thought that I will explain the food security also a little bit in more depth so that we can take up the other issues along with. So when we talk about food security, it is technically access of physical and economical access of food in sufficient, safe, and nutritious food. So this is should be available to every individual on this earth. So it is, maybe we can say that India is producing a lot of food, but is all food is available or accessible to every citizen of India? The question is definitely no. So that is why food security is not only production of enough quantity of food, but making it available or accessible to every individual is also important. So that under the broader term of food security, these are the minute things that we need to look upon. So as a policymakers also, normally what we talk about is how much production we had this year and what was the increase in production that we had over a few years. That is the only focus. But access of, say, sufficient, safe, and nutritious food is also an ultimate goal of food security. And that is why the issue of food security is very complex, and it is having multiple dimensions into it. So how to ensure food security? It is, first, of course, we have to increase the production so that you can make food available to all. Then second thing, we have to make the food access to everyone. And third thing, you should also improve utilization of food. Utilization means better food distribution, food processing, preservation, and storage. So if we don't preserve well, if we don't store it, we don't have proper storage, we will lose a lot of food that we are producing. So these three are considered as pillars of food security. That is increasing food availability, access of food to every individual, and also improve upon for its utilization. Okay. Now, as our DG sir has also recently in his message, he has said that we need to increase our food production by about 70%, but there are different figures that have been put by different statistics. And this is one particular statistics that has shown that by 2050, we will be having a population of around 10 billion globally. And the food gap by the time, if you look into the current trend, we will have a food gap of around 56%. So this 56 or 70, it is based on certain estimations. What you can see is that around 60 to 70% more food we will be requiring to feed that population by 2050. Somebody will say we are producing enough food. So that's true. But if you look into the global scenario, you will find that there is still a gap. And that gap is mostly due to the food production scenario in the low income countries, particularly the African countries and so on. Although some countries, we are having good amount of food production and we are happy about it. But the question is that, is it sufficient to meet the food demand globally? Because we have other countries who are producing very less food for their population. So this figures justifies that statistics in a sense that the middle one is the curve is the current rate of food production, and the gap that we will be requiring by 2050 with the current food production is mostly due to the slowness, or you can say that a lesser amount of food being produced in the low income group countries. So that is why we need to have a concern over food security at global level. As an individual country, probably we may not face that challenge so soon, but we have to, because in the current, even the war scenario, you can see that there are shortage of food in some part of the world, and India is helping other countries to provide food, particularly countries like Afghanistan and so on. So that means we cannot let human being to die because of unavailability of food, even if they belong to a different country. So that is a moral responsibility of every individual at the global level. So if we have an excess food production, that will definitely have better make food better availability to those countries who are unable to meet that demand. Along with the food security, we also need to address another important concern, that is malnutrition. Currently, global malnutrition level is estimated to be around 11%. So it is not only the calorific value of the food, it is the nutritious food that also forms a very, very significant part of food security. And we need to address this because a lot of people still are having, facing malnutrition. Now, data says that if we just look back to past 50 or 55 years of agricultural production, we have made a lot of progress. Here you can see that in case of cereal production, we from 1960s to say mid of last decade, we were able to increase our cereal production by 220%, vegetables by 425%, other roots and tubers crops by 85%, and fruits and including that 294% or around 300%. So what we did is that we, when we talked about food production, we have tried to increase the level of production of the crop. But what we did not emphasize is that increasing the cultivable area. Because again, if you just see that today's current breeding programs in agricultural crops, breeders are breeding crops for disease resistance, some nutritionally improved cereal grains, or you can say that they are working on abiotic stresses and so on. Hardly they are focusing on increasing yield. Why? Because the potentiality of maximizing the yield has already been achieved. So there is very less scope for crops like cereals to enhance productivity from a single plant. We can say that it has already hit the ceiling. So what do we do then? The only possibility is that we need to increase our land area, which out in last 50 years, it is hardly 20% of land area has been increased. So a committee was set up by the UK Royal Society to look into these issues under the leadership of Professor David Balcombe. Probably his name is more known for RNAi interference like phenomenon. He is the pioneer worker in that particular area. So the committee advocated sustainable intensification in agriculture, where the crop production should be on the basis of resisting towards stress and diseases, so that external chemical inputs should be reduced, produce consistent yield using renewable inputs, avoid depletion of minerals, diversity and natural capital, and so on. And the committee suggested that to achieve this sustainable intensification in agriculture, what more is required, what the scientists need to do is that to advance researches in the field of genomics, systems biology, microbiology, and cell biology. So my talk will be focused on that particular aspect, that is microbiology, in using of microbiology in agriculture. So when we talk about microbes, it is there in every part of the food chain that we have, right from germination of a seed to consumption of the particular food product until it gets digested. Everywhere microbes are involved. So you can imagine the role the microbe can play in our day-to-day life. Because without microbes, probably we will also die out of hunger. So it's time to understand and manipulate the microbes to develop novel strategies that may contribute to improving food security. Now it is said that healthy soils are required for healthy plants, but now the slogan is increased that healthy plants are also required for healthy human beings. So that means from soil to our health, it is all correlated. We are directly related. Earlier, we were thinking soil health only in the context of plant health. But now we understand that plant health is also important for our own health. Say, for example, you may consume certain things, but if the product per gram of the product is nutritionally not rich enough, so you can fill your stomach, but you are not getting the desired nutrition out of it. But a healthy plant, it's not in terms of its biomass, but when you talk about a healthy plant, so nutritionally also it will be optimized because of the presence of the microbes that surrounds the plant. And finally, if you consume certain microbe raw at the plants as a raw, it also influences our gut microflora. So altogether, I will not go into much detail into this topic, but what I'm trying to relate here is that everything is important for us, and everything has a common linkages, that is the microbes. Whether it is present in the soil, whether they are present in the say plants, ultimately they are benefiting us, our health. So that means we should not look soil as a separate entity, plant as a separate entity from our own health. We should take care of all these things. So microbe offers solutions to food security by helping in 3 different ways: reducing food spoilage and outbreak of foodborne illness, increasing food production, and creating new foods. So I will be focusing on the middle point, that is increasing food production, leaving the 2 other points aside for the time being. So food basically comes from plant, and we consider plant to be a superorganism. Can anyone say why plant may be a superorganism? It is mostly because plant harbors a diverse amount kind of microbes, either externally or internally. In externally, we call them epiphytes, and those microbes that reside within the plant, we call them endophytes. So what do you understand by looking into this particular picture? It shows that the microbial community in the same plant in different parts are diverse. It's not that a single or same plant harbors the same kind of microbes from root to, say, leaves. The root endophyte population, there may be some microbes in common, but overall, this is a classical example where the root, lower leaves, upper leaves, flowers, even the fruits, they all harvest different kind of microbes. Some may be similar. So that means this again gives us a lot of thoughts to think that why this diversity is there in different parts of the plant. First of all microbe will not go into the plant system. Plant will filter them to so that only certain microbes are welcome to the plant inner tissues. And among those also, they occupy, different microbes occupy different parts of the plant. And definitely it is not a randomized structure. It is definitely based on certain biological significance. We are yet to understand as a human being what say role these microbes play in different parts of plant, but probably with more research advancement in research, we will come to know how different microbes are performing different activities in different parts of the plant to make a plant more healthy or more productive. We human beings are very selfish, so we see everything from our own benefit point of view. So we also discuss things in that particular angle. Now, if you talk about the plant genetic potential, we know that plant harbors genes from around 27,000 to around 1 lakh genes, from Arabidopsis to wheat and other falls in between. But plant genetic potential is not the plant chromosomes itself. It is also plant genome can be considered that it is a combination of the plant genome as well as the associated microbial genomes. Because these microbes, they are so intricately associated with the plant system, they behave just like a part of the plant. And say, for example, the microbe which is within the plant system, say, for example, endophytes, if the endophyte is producing an antibiotic compound and the plant is getting benefit out of it because it is killing some of the pathogenic, say, bacteria or fungi. So what we can see in a broader sense is that this microbial gene is also helping the plant to survive in a particular stressful condition. Here we are talking about a plant pathogen. So that is why the plant genetic potential can be expanded to the associated microbial genomes as well, because plant get benefit out of that genome as well. So rhizosphere is the most dynamic stage in that context, because it harbors a diverse type of microbes, and it is also get changed depending on certain factors. So broadly, the rhizosphere microbes can be classified into one group that have a negative impact on the plant. That means they can destroy the agricultural production. And the other group that produce, that have positive interactions with the host plant, which can basically restore or sustain agricultural production. So there are certain microbes which are very neutral in sense, or rather we can say we have not yet identified their role. But broadly, the microbes can be grouped into two groups. One is harmful, another one is beneficial. Now again, agriculture is not a static area. We have from wild cultivated plants to the current cultivars, we have made a long journey. And what we did is that we have selected plants based on certain traits or characters, and we have also used those characters for in our breeding programs. And finally, the modern cultivars are available. But what we see in general is that the wild relatives were normally, they had lesser foliage, they were taller in size, but had a very big root system. But modern cultivars, they have more foliage, smaller root system, and that is why we need to put a lot of external fertilizers so that we get more yield out of that plant, because the nutrient absorbing capacity of these plants are already reduced because of the smaller root size. So that means this is one, say, phenotypic characters of the plants. But what actually happening in the rhizosphere is that if you see the rhizosphere microbiome, and you will see there is an abundance of bacteroidetes. But in modern cultivars, the rhizosphere microbes are mostly dominated by actinobacteria and proteobacteria. So that is the major difference from the wild relatives with the modern cultivars of the same species. Why it is so? Because we know that plant root actually releases around 25 to 40% of the total photosynthetic synthetic products, that is carbon products. And this carbon product serves as a food for the rhizosphere microbes. And this rhizosphere microbial population varies because of this change in the exudation pattern. And this exudation pattern is again governed by the plant genetic characters. Even say, for example, you take 3 wheat varieties. Wheat is the same crop, but these 3 different cultivars. 3 different cultivars means it has genetically, in some areas, it has some different genes that harbors. All these things makes the plant total exudation pattern is different. And because of that, we get different microbial community in the same species of different cultivars. So in the same soil, if you grow 3 or 4, any different type of crops, you will, and then analyze the microbial community, and you will find a difference in their population structure. It is mostly because of their exudation are different. Although the soil is same, microbes, the resident microbes are same, but still we get a different community in the rhizosphere because all different plant species, they exude out different types of root exudation. Then again, in a single plant, in the single soil, the microbiome can get changed if the plant comes under certain stress. At that stage, what plant do? The plant also ask for help, or they can cry for help, in a sense that they change the pattern of exudation so that certain beneficial microbes can be more recruited in a greater amount. These are all established facts. Say, for example, under certain stress, like pathogenic stress, like Pseudomonas syringae, the plant exudes out more malic acid. And this malic acid then serves as a chemoattractant for certain bacterial species like Bacillus subtilis. And this Bacillus subtilis has the ability to suppress the infection of Pseudomonas syringae. Similarly, plant can, under say low temperature or cold stress, plant can increase the production of strigolactone. That will attract mycorrhizal fungi like glomus, and ultimately the plant will be able to tolerate this low temperature because of recruiting this new microbe through the chemical signal strigolactone. So there are numerous examples that has been now people have documented how under stress condition the pattern of exudations get changed, and because of this changed exudation, they actually invite new friends which will help the plant to survive that stressful condition. So this is what I was talking about in Arabidopsis. When it was challenged with the pathogen Pseudomonas syringae, they have documented that malic acid transporter genes are activated that increase the malic acid biosynthesis and exuding out through roots. It recruited the biocontrol agent Pseudobacillus subtilis, and it triggered a induced systemic resistance, and that has ultimately suppressed the infection by Pseudomonas syringae. So this is a study that I was part with, and it's a global level study where we actually sampled the chickpea nodules, and we wanted to see how this nodule is a very, very important part for biological nitrogen fixation in legumes. So we just wanted to see what is the microbial dynamics in the nodules in different parts of the world. We have considered few parameters like soil type and the latitude. So we found a correlation between these two. Where the soil type and latitude is the same, we find mostly these nodules are occupied by a similar type of bacteria. Whereas when it gets changed, the pattern of nodule gets, the microbial composition gets changed. That means same chickpea, same nodule, which is a very, very tightly regulated phenomenon took place in that nodule, that is biological nitrogen fixation. But this, even this tightly closed environment also, we get different types of microbes based on certain other factors like soil or latitude. This is another study that I am quoting from other labs, where they have found that in the plants, the endophytes and the rhizosphere microbes, and they have analyzed and they have found that endophytes, their diversity is lesser in comparison to the rhizospheric microbes. And the same plant, same soil, if you alter the condition here, what they have altered is the water stress. In one set of plants, they have created water stress, and they have found that the diversity of the endophyte doesn't get changed much, but the diversity of the rhizosphere microbes, they get changed drastically. So that means even the plant is same, soil is same, but if the environment is here the environment is the soil environment where the water level is different. In one set, the drought was created, and there is a change in the pattern of the microbes that is associated in the rhizosphere. So that means it is studying microbiome is not a very simple task. This is what I want to trying to emphasize here. It gets changed with different in condition. Whether we can keep the soil intact, we can keep the plant variety intact, still it is very, very dynamic. So how do we catch them? How do we actually follow the pattern? What should we trap out of this information? What should we conclude? So some answers lies here. So say, for example, a plant is harbored by 1,000 different species of microbe, but all of them are essential to the plant? This question was not served till some years back. So what they did, they experimented by using tomato as a model plant. So they have harvested microbes from the leaf of tomato plant, and they have taken the seed of that tomato and planted in the next season. And they have inoculated the same microbe that was harvested in the first year in the second year. Again, they have harvested that microbe from the second year, and they have done it for 4 subsequent years. After 4 years, when they analyzed all the populations in the 4 generations, they have found that out of the total microbes, there is hardly 25% of the total microbes which are actually found in all the 4 generations. And rest 75 is actually passengers, you can say. It is seasonal fluctuations. They have come to the host surface by chance. So that means these 75%, they are not of our interest. Only this 25% is our interest because they are closely associated with one plant species, and they are associated in subsequent generations as well. So this is how we need to, this is what they termed is a core microbiome of a plant species, CORE, core. So we have to, when we talk about microbiome, now we know the idea of core microbiome. So we should chase this core microbiome for our commercial benefits. In terms of ecological fitness, it was also seen that the core microbiome of a plant species, they are ecologically fit in diverse conditions as well. There is little change in this, but still a majority of the core microbiome, they are associated with the plant in diverse environmental conditions. Here the experiment was taken in hot places versus cold places, and they have found that even in the hot or cold, whatever the temperature, outer environment it may be, the core microbiome was mostly stable. It's only the passengers microbiome, they form two components distinctly, and they are not of our concern. This is another classical example how the dynamics of microbiome get changed in soil under natural condition. These are two plots side by side. They have experimented using wheat as a crop and Gomminomyces graminis, a pathogen of wheat, as a pathogen. Initially, both the plots, they are adjacent to each other. They were devastated by the wheat crops were devastated by the microbes, pathogen. So in one plot, what they did is that they have rotated wheat with other crops. And in one plot, in the right one, they have not rotated the wheat crop. Rather, they have allowed the wheat to die initially, but they have used wheat as a single crop over years. After around 12 years, what they have found a situation of this level. That means when you have rotated the wheat crop with other crops, you are actually disturbing the soil microbiome, because different plants are actually changing the microbiome potential. But in the second plot, the plant cried for help, and slowly, a particular amount of microbial community got built up, which ultimately started helping the plant by suppressing the pathogen. So you can see hardly any disease in the right panel. And in the left panel, you can see that that particular plot is again suffering from the disease. So that means the plants, when they cry for help, we should also give them time to rebuild that particular trait, that is reconstructing the microbiome of the soil, so that at one point of time, these microbes, the beneficial microbial population will be higher in the soil, make the soil suppressive for the pathogen. So nature can also do that, but only thing is that it takes a lot of time. And we as human beings or as farmers, we cannot wait for this long to achieve this goal. So what we can do is that we can identify the core microbiome, use it as a commercial product, and apply it so that we achieve this zero disease condition. So we started with some microbes like Trichoderma, Pseudomonas fluorescens, and Rhizobium, and we used the model chickpea for experimenting on it. The normal approach is to see the antagonistic activity of the biocontrol agents. That means whether they have any potential to suppress the pathogen. We selected some very potential strains. And when we tried to see that whether this, say, Trichoderma is compatible with Pseudomonas fluorescens or Rhizobium, we have found that some are compatible, but almost 50% of the Trichoderma strains are incompatible with the Pseudomonas and Rhizobium. The second point you can see here. So that means selection of microbe is also very, very important. All are Trichoderma strains, but around 55% are positive in terms of they are compatible. Around 45% of the Trichoderma are incompatible with the other beneficial microbes like Rhizobium and Pseudomonas fluorescens. So we have highlighted that particular point in the article that we published in Soil Biology and Biochemistry, where we have shown that any consortium that we develop first we should have a compatible strains in hand. Trichoderma suppressing Fusarium, very good in one way, but if it is not compatible with Rhizobium, so it may have some impact on nitrogen fixation. Or if we are mixing with, say, Pseudomonas fluorescens, Pseudomonas fluorescens can even suppress the Trichoderma. So what is the fun of using that kind of strains? So this has to be, because when we were using this consortium, when we were referring different, say, literature, what we find that people will take a strain from this lab, another strain from another lab, and they will simply mix. And there are reports that people have reported reduced plant growth. So that made us more curious why people are reporting reduced plant growth when they mix it. Because individually, both the strains, they performed very well. So then we experienced this, and we have highlighted this particular aspect in this particular journal. So when we applied the compatible strains, we could found that it was best performing better than the individual strains, or when we mix two strains together. But when we mix the three that are compatible with each other, complementing in different things for plant growth and development, we found best result in terms of, say, ROS reduction in ROS activity, catalase activity becomes higher, or even the chitinase production become more in the triple combination of the microbial strains. This is one physical evidence. You can see that the bottommost panel D7 is the combination of all the three microbes, whereas A is the untreated control and B is the pathogen treated control. So you can see the amount of lignifications that takes place. It is highest when we went ahead with the compatible strains of three different microbes. Even they have performed better than D1, D2, D3, D4. These are individual treatments as well as combination of two strain treatment, but D7 was best among them. Then coming back to the change in climate. So Dr. Chaudhary has also mentioned that soil salinity is becoming a problem in agriculture right now, because soil salinity is affecting the productivity of the crop plants. But what we don't know is that in this saline soil, how these beneficial microbes are behaving, or what is happening to the pathogenic microbes, because we are talking about only the plants. Plant productivity is going down. But what is happening when you talk about soil, that it's mostly, we should talk about the soil microbes as well. So what we did is that when we started screening, we found that in chickpea, when salinity is increased, there is extensive colonization of a pathogen like Fusarium. You can see in the bottom right panel, where at the root zone, extensive colonization of the pathogen taking place when we have increased the NaCl concentration in the, say, growth medium. So what does it indicate? It's not only actually affecting the plant growth and development, rather it is promoting the growth and development of certain pathogenic species like Fusarium. So this is a very, very contrasting thing. And combination of these two, you can imagine salinity alone is affecting the plant productivity, and it is promoting the activities of harmful microbes like Fusarium. So it will be a deadly combination, and hardly we can expect any yield from such fields in later if pathogens population like Fusarium keep on increasing in these soils. So data suggests that the fungi, the Fusarium, their mycelial growth is increased, and sporulation is also high. But there is a ray of hope. When we introduced a strain of Trichoderma, you can see from 100% mortality, we can bring down the mortality level below 20% or up to around 20 to 25% when there was a deadly combination of both salinity and say Fusarium. So that means it is, the model says one thing, that yes, we are going to face a very severe challenge in saline soil, but we have options to deal with that also. in terms of using microbes like trichoderma. We have used trichoderma, so I am just giving data for trichoderma, but there may be other microbes who will be performing in the same way. So what next we did, we tried to see the microbial community structure in the rhizosphere of such soils. And we started with the phylum data, where we have found that when we introduce trichoderma, then what we see is that we have seen, is there any pointer? Okay, yeah, you can see. You can see that this is TP, means trichoderma and pathogen. Okay, thank you. You can see here, it is increased, this green portion. What is this green one? This is the phylum Crane Archaeota. Okay, it's an archaea. Dr. Chaudhary was mentioning that we have to focus on that particular protozoa and archaea. So this is what we have found. Then when we dug further into the data, then we we came across the genus level data. And here it is showing that this TP, that is trichoderma inoculated in presence of pathogen, you can again see there is a tremendous increase in this green patch, which indicates the genus Candidatus nitrosophora. Okay, so this is what we have got in hand. Second situation was salt, salinity. In this case also, TS, trichoderma 1, it was introduced in presence of salinity. Again, there is increase in Crane Archaeota, the same strain of trichoderma. And in case of TS, trichoderma plus salt, this again green patch increase, and this is Candidatus nitrosophora. So this trichoderma is doing something in the rhizosphere, which is basically reducing the total level of proteobacteria. जो हमने बताया था कि आपका जो modern plant genotypes है, their rhizosphere is dominant by proteobacteria. But this trichoderma is doing something reverse. तो ये थोड़ा confusing था हम लोग के लिए कि what is happening, because it is going against the trend. So what is there? We tried to understand. Then since nitrosophora is not a common genus for me, so I just searched literature, and then I came out with the information that it is basically an archaea that oxidizes ammonia. So that means when you introduce trichoderma, the population of this particular archaea goes up and it oxidizes ammonia. So ultimately ammonia oxidation lead to nitrate production, and nitrate is taken up by the plants. So we wanted to see whether plant is taking up that nitrogen that is being converted from ammonia to nitrate. Then we selected one gene, that is NRT 1.1, which is a very good nitrate transporter in the plant. In those plants where we use trichoderma as a seed treatment. And we have found that in presence of trichoderma, the activity of NRT 1.1 is increased. So that means this particular trichoderma, it is increasing the nitrate uptake machinery in the host plant. At the same time, it is also influencing the soil microbiome by suppressing the proteobacteria or increasing the archaea species like nitrosophora. So we can use this particular trichoderma as a hub microbe. This is an example I'm just quoting from a different paper that was published in Nature Plants, where they have given the concept of core microbiome behaving as a hub microbe, hub for other microbes. So here, trichoderma is one such microbe. In our study, we have come out with, it can be used as a hub because it will probably develop a network of other beneficial microbes along with it, and this altogether, this particular new community will actually help the plants to grow better in stressed conditions. So this is what actually I wanted to convey to you all, in a sense that the microbes has tremendous future. And we have, with our own experiments, seen certain aspects of those, how a single or few microbes can actually transform the agricultural system into a productive one, not only increasing yield, but also, I have not presented the data right now. We have also seen that the antioxidant value of chickpea edible parts also get increased when you use microbes like Trichoderma. That's the strain that we are using, T42. So it increased production, and it also increased the nutritional value of the produce that we actually consume out of it. Second thing, it also helped the plant to survive the stressful conditions like pathogenic stress or abiotic stress like salinity. This is our model. It may have more impact on the plant. We don't know because we have not conducted that studies. So this is the future. We should chase this core microbiome. We can start with few microbes that are giving benefit to us, and then we can build a core microbiome out of from the data that we generate, and that can be used as a potential say product for enhancing plant productivity, along with helping the plants to cope up with stressful conditions without taking help of any synthetic chemicals. So we have worked on several microbes, like from Streptomyces to Trichoderma and combinations. So I will not present all those data. So the roadmap that I suggest for us to go ahead is that we should identify the core microbiome, either taking help of the conventional culture-based technique, or say modern techniques like use of bioinformatics to develop this core microbiome, and we can finally have a product for every individual crop or every soil type or every stress condition. So we may have designer microbiome for an individual crop like tomato growing in a, say, high altitude area, saline area, or whatever it is, that can be addressed through this core microbiome. So these are my students who actually conduct the research, and I'm happy that some of my research activities I also did with Dr. Anukul, who will be the next speaker, and he'll be also presenting some very fascinating data to you all. And the funding that mostly we received from Department of Biotechnology, Department of Science and Technology, and Indian Council of Agricultural Research. So I acknowledge their help in getting those data. And I thank you all for your patience hearing. Thank you very much. So if you have any questions, I'll be really happy to answer some of your questions if I have any answer for that. Yes, please, sir. Technically, I will give you an opposite reply. Due to our own biasness, I started looking at the mesorhizobial species, because this is the species that actually help in nitrogen fixation in chickpea. But to our own surprise, even that mesorhizobia species population has gone down. So that was initially we thought that the data is wrong, but later on we started working on those, because these are repeated experiments. We have got the same thing. So we said that now data is correct. So let us try to understand what is happening. So under stressful condition, or probably there's another explanation that I have, is that because we did this experiment in our own BHU farm, which is already rich in nitrogen. So we know that the nitrogen fixation takes place only when there is nitrogen level goes down, because these fields are already bombarded with so much of urea and DAP. I don't think that may be one aspect that what we are actually trying to relate with, why we are getting so much less in, and that mimics the farmer's field also, because even if it is in BHU, if we are applying a recommended dose, farmers are using more than that, in particularly urea and DAP. So I think this is an experiment that mimics farmers' fields as well. So we are getting a declination of rhizobacteria, rhizobium species, but increase in archaea-like population.
Right, crane archaeota especially. you
Ukraine
mentioned.
archaeota in our
Right,
case that we got.
Because
Yeah.
few years back, we were dealing with the Enterobacteriaceae group also.
Okay,
In the rhizosphere and in endotrophic mode of nutrition,
okay.
endophytes.
Mhm.
So Enterobacteriaceae group usually locate inside us, intestinal microbial flora. Enterobacteriaceae, you may find out the same. It is opportunistic in the rhizospheric scenario. also.
Yeah,
So,
definitely. Yeah.
So I was wondering to find out either the alpha proteobacteria, beta, gamma, delta, epsilon. So it is beta one, I think, if I am correct, or the gamma one, because Pseudomonas also much reported over there.
Exactly.
And thank you, sir. Thank you.
Yeah,
I said
thank you.
Mesorhizobium, very good.
Sir,
Thank you.
Thank you for excellent presentation.
Thank you, sir.
A lot of new information.
sir, thank you.
Very
so
well
much.
done.
Thank you, sir.
I've got two questions.
Sir,
One that arbuscular mycorrhizal fungi,
Mhm.
when they enter the root, they mostly stay in the exocortex
Yeah,
and sometimes go inside deep. But they never invade the xylem and phloem or pericycle, what you call it. There's one thing. What is the reason for it? They have the enzymes to liquidate that. And the same thing happens with the endotrophic bacteria, fungi. So these are two things which are bothering me, which I could not get the answer so far. What is the reason why they stop there? One.
Yes, sir.
Second, they live in the root of the plant, but they never go up in the higher side. What is the message they get? Stop here.
sir, I think for these questions, you are the best person to give answers, but I will try to put one logic from my own experience with the endophytes and their compatibility with, as I have mentioned, that different plant parts are occupied by different species of microbe. So that means there may be also a competition between mycorrhiza to go further deeper into the plant tissue with the resident endophytes. That may be one reason. I am not sure, or neither I am expert in that area. As sir has said, that mycorrhiza has the enzyme to go to that level, but they don't go there. Probably they face a stiff competition with the resident microflora in the those tissues. That may be one explanation that I can guess, but truly I am not an expert in that area, so I don't have a proper answer for you, sir.
Yeah, when I made a book on mycorrhiza,
Okay.
along with Professor Hock in Germany, I opened a question
Mhm.
that we call VAM, vesicular arbuscular mycorrhizal fungi.
Right.
But I dismissed the word V. I said it should be AM only.
AM.
AM fungi. There were a lot of controversies all over the world I
Yeah.
received that you cannot change the terminology being going on for the last 200 years. My argument was that vesicles are very common in fungi, but arbuscules are very unique to
Unique.
mycorrhizal fungi.
Yes.
Nowhere else. Now it should be AM. Of course, after some, but I published all those things, controversies.
Mhm.
But today now people are realize that it should be AMF. In the
Definitely.
same book, I also raised these two questions, but so far I did not get reply
Yes.
from anyone's word. So this is a very open question in my mind.
Definitely.
What is the limiting factor not going up? Not a limiting factor not going to pericycle.
Exactly.
Okay, thank you.
Thank you, sir.
Thank you for this so insightful lecture you have presented. I just had a query that you suggested. You came up with this idea that we can be having the customized this consortia of the microorganisms to address like region specific Like some areas may be saline, some areas may be having drought conditions and all. So how do you suggest that we can address the variety? Like we are taking the example of tomato as in your case. So because the variety of tomato or any given such plant, tomato we are talking because that is a model plant you have considered. So how do we address that thing, that differentiation
Yeah.
between the variety?
Definitely. Thank you for the question. It's a very excellent question in a sense that we also need to look upon various aspects like plant genotypes, as I have mentioned. But when it comes to a stress, then even if the literature suggests, I have not worked on those areas, even the plant genotypes remains the same, the microbiome actually gets changed according to the stress. So that means we have the scope of developing a microbiome, say for example, drought situation. So when drought will be there, a specific pattern of microbiome gets built up. So that type of data suggests that we can actually develop, because these are stresses. So we can, and these are built up around that same plant species. So that means plant is getting benefit from this newly built up microbiome.
Probably the root exudates play a very important role because they are helpful in recruiting that kind
Exactly.
of rhizospheric microbiome.
In normal soil, there is a different level of EA, but in a stress soil, even the susceptible plants, their exudation pattern is getting changed. And ultimately, this newly built up microbiome gives us an indication that this particular microbiome will help this plant species to grow in stressful conditions like stress or salinity. Yeah.
Thanks a lot.
Thank you very much.
First of all, thank you, sir, for your nice presentation.
Most
I always enjoy your presentation because you always use latest references and interesting study in your presentation.
Thank you.
I have one answer of Verma sir question about the limitation of mycorrhiza, why it's not enter up to the high height in the plant.
Okay.
So I got an opportunity, sir. I worked with Professor Marcel in Switzerland. He is one of the renowned scientists in mycorrhiza field. So I can suggest one thing. He discovered that the mycorrhiza is not only responsible for entering the plant, it always related with the mycorrhiza helper bacteria that helps to mycorrhiza for entering in the plant. And he found that at some extent, those microbes get lost inside the plant, which are the helper bacteria of mycorrhiza. So once mycorrhiza enter in the plant, that microbes get lost, maybe because of the endophytic bacteria of microbes that you said that at different microbes is there. So maybe that would be the reason, because mycorrhiza helper bacteria does not work inside the plant tissue. They only works in the outside the, we can say the mycorrhizosphere or the rhizosphere. But once mycorrhiza get inside, the mycorrhiza helper bacteria does not work properly.
Yeah.
So maybe that is the one of the reasons, sir. Yeah.
Yeah. Yeah, this is quite possible because it is also going inside the plant tissue with taking help with other microbes.
Yeah, we published one chapter also. We both mycorrhiza helper bacteria for your book, mycorrhiza and pedogenesis earlier also. So nice interaction with you, Dr. Sharma. Please. Dr. H.B. Singh, sir.
Yes, sir. I have one query, Dr. Sharma.
Yes,
Sure,
sir.
please.
Compliments for a wonderful presentation.
Thank you, sir.
To my knowledge, I know only one product in the country launched by C Biotech, the Dr. K.R.K. Reddy. So do you have any idea what kind of microbiome for which crop the product is there in the market? I don't have any idea. I was trying to find out from the literature, but I couldn't get that. So
Sir,
that is the only company in India
Right.
launch a
Right.
microbiome based product. I do not know that product is registered in pesticide, biopesticide, or biostimulant. No idea. And I was checking with ADG Plant Protection. Then he said it is not registered with the biopesticide. So anyone who can answer this, not taken. Dr. Reddy is there, Dr. KRK Reddy.
Okay, yeah, actually, sir, well, I don't have any answer for that because I don't know exactly for which crop it is being used. But as far discussion with you and the CIB people, we know that probably India is going to have the license for consortium products.
Yeah,
So that is,
I will be
used-
tackling that in my presentation. Yeah, I will talk on that.
Yes, yes, that will be, that
Yeah.
is one good news, I think, coming in our way, that
Yeah.
we will have the regulatory clearance for commercializing this consortia as a product. So far, it is only for single microbe that was permitted for commercialization.
Still, a lot of things are going on in CIV, and till date, no microbial consortia has been registered. But they are struggling hard to find the guideline, how to do that.
Yes, sir.
So,
So we'll be
so
looking
hopefully,
forward
they will
for
be
that
doing that.
day.
Yeah, hopefully they will be doing that. So I skipped my office and came back to home now.
So thank you, sir.
So,
Thank you.
yeah.
Second half at 2 p.m. Dr.
Yeah.
Singh?
Yes, sir. Yes, sir. I'm comfortable anytime now.
Okay, at 2 p.m.
Yeah.
We shall see around Professor H.P. Singh with us. Few more questions we have for you,
Okay,
Dr.
okay.
Sarma.
Thank you.
Gaurav? Gaurav and Sahil, stand up, please. So among students who would like to ask questions, raise your hands, please. Okay, after Gaurav. you-
Sir, I'm Gaurav Yadav.
Thank
I'm
you.
PhD scholar under the guidance of Dr. D.K. Chaudhary.
Okay.
I have two queries.
Mhm.
One is, as you shown that the plant in different region of plant have different microorganism. So it is vice versa, or it is dependent on how the plant select the microorganism, how it will let inside its system.
You see, plant is also when microbe has co-evolved, and a single plant species is also been cultivated or grown in other part of the world. So that means plant has the ability to accept diverse type of microbes, but not all. So second thing is that suppose we are growing a plant in, say, in Indian continent, we have a specific around kind of microbiome that is present in Indian soil, but some microbes, they may be of diverse species that is in, suppose, New Zealand. So but the plant is grown in both the places. So the plant has a basic mechanism to accept certain microbes. Based on the resident microbe of that soil, it will actually intake, or you can say the filter those microbes into its system. And the nodule study has given us that idea. The soil type also support certain group of microbes. I'm not saying that other group of microbes, they will be completely absent in certain cases, but their population will be very, very less. But some, based on say soil pH, some microbe will be more dominant in a particular pH. If you certainly increase the pH or lower down the pH, the same soil will show dominance of other microbes, microbial species. So that is why soil type is one of the factor that supports the dominance of a particular species of the microbial strains. And once you have the dominance of certain few species, definitely if they have those microbes that can go inside the nodule, it will go there. This is actually the plant soil actually determines the microbial dominance. It is not presence or absence. It is the dominance of certain species in a particular soil type.
Thank you, sir. And
Okay.
sir, my second query is that
Yeah.
if we are interacting two organisms and they are showing like interaction in vitro study, but if we subjected them to the in vivo, but they are not showing that much result, is that possible?
Quite possible. Normally, when we isolate microbe, again, it is the dominant microbes, they come at first. So that means from that particular kind of soil, that particular microbe has dominance over there. So that is why you are able to isolate them very quickly and easily. Second thing, it is not a one-step process. Even if we have a compatible group, we have to test it in pot, we have to go into microplots and then field level study. So the process is quite long, but what I showed is that the initial thing where we commit mistake, we simply randomly we mix microbial strains. Even individually, they are excellent microbe to provide some benefits to the plant. So when we combine, we also need to see whether they are compatible with each other at the very beginning. Then this has to go under different level of testing from in vitro to in vivo level. Then only we can actually recommend this for commercialization or for farmers.
Thank you, sir.
Thank you.
Alongside, I have one thinking, if you permit.
Yeah, definitely.
Dr. Sharma.
Yes.
The 25% core microbiome you talk about, does it relate with the Koch postulates? You know,
Yes.
we may think in that way.
But actually, core microbiome concept is little new in a sense that we don't know too much about that. So that data, that study has first spoken about that all microbes are not essential. There is a certain percentage of the total microbiome. They are found associated with that plant species over several generations and in different areas. So that gives an indication that these microbes are probably very essential for this plant. But again, these microbes will not be present in other plant species. It is not ruled out. Or rather, we don't have data to suggest that. So probably some of them are also part of core of some other plant species as well. So that will be an interesting study if somebody conducts the how much percentage of core microbiome of, say, tomato is also core for potato or wheat. So we don't have data right now to, but I believe that definitely they are not truly one species specific. They are specific to that species, but some of them will also be part of the core with related species of plants, maybe. Yeah.
Good afternoon, sir. My name is Sahil Devnath. I am currently pursuing BSc Honors Microbiology.
Okay.
Sir, I have a single query.
Sure.
The rhizobium bacteria that you mentioned in your lecture that are present in the root nodules of the leguminous plants, which helps in the fixation of nitrogen, and they convert nitrogen into nitrates and nitrites for the plant purposes. So, sir, I have a question like what special characteristics they have that makes them special, and what kind of characteristics makes them helpful in performing this function?
Okay. You can sit down. First, the rhizobium, you probably know that they have NIF genes to fix this atmospheric nitrogen. So that makes them unique in that sense that there are other bacteria that also harbors NIF gene, but this rhizobium, they are actually symbiotically, you can say that they develop nodules and they fix nitrogen only inside the nodule, not in free living state. So during the process of evolution, plant and microbe evolved in a way that, say, for particular legumes, they have this nitrogen fixing bacteria in a sense that probably we don't, I don't know how to explain it properly, but this is part of their evolution that they have come or they have evolved together so that, you know, you can say that why other free living nitrogen fixing bacteria like azospirillum or they don't go into the plant root nodules because plant don't accept them, legumes don't accept them inside the nodule. So that means this has the mechanism, they have evolved together to develop this particular system of nodulation and then fixation of nitrogen within the nodule. But rhizobium normally actually fix nitrogen in the form of ammonical form. So it has to get converted into nitrate and then plant uptake nitrate as a. So what we did is that nitrate transporter genes we tried to look upon, and we have found that trichoderma is actually, it's not a nitrogen fixer, but it is actually stimulating the nitrate uptake genes in the plant. That means the plant is now more efficient to uptake the nitrate from the soil. So this is the correlation that we have obtained, but this is a very different mechanism from nitrogen fixers like rhizobium. Okay.
So number of questions. Yeah, one more, please.
Sure, sure.
Yeah.
No, we'll be happy.
Yeah, thanks a lot. I'm not a plant biologist, I'm a immunologist, but after listening to a couple of discussions, I thought of asking one question.
Sure.
So please excuse me if I'm wrong because I don't belong to the field. So I refer to the crop rotation. This crop rotation is a strategy of producing more and more hybrid vigor crop. So do you think that this crop rotation mechanism for cultivating variety of the plant in the same area is correlated with the microbiome in the soil? So how in the crop rotation mechanism or experiment, the discrete microbiome would actually enrich the growth of plant? And if this is a case, can this become one futile experiments to understand the diversity of the microbiome?
Definitely. We can take two samples in that sense, the rotated crop versus a virgin soil like forest soils. And you can see the stability of the microbiome. In forest lands, we don't get too much of disease plants because the nature has taken care of those things. But in agricultural setup, we have, for our own benefit, we have promoted monoculturing of crops. That has created a lot of problems. So definitely when we go for crop rotation, so the same microbiome actually population doesn't build up according to one plant species. So it builds up in one season and it gets destroyed, and a new population it builds up with the subsequent crop. So probably that is another reason that we don't have a stable microbiome in agricultural soils to take care of the stress issues, whether it is biotic or abiotic, because we are continuously building and destroying, we are not making them stable. So that is definitely one aspect that we need to focus on. But as I have mentioned earlier, we as a human being, we see our benefit first, then we talk about the environment and other things. So this is very valid. And it is, I'm truly convinced, although I have not worked on that data, but I'm convinced that we are not able to building a stable microbiome that is actually healthy for the plant, because with change in crops, we are destroying the initial microbiome and building a new, or starting to build a new microbiome. And then end of the season, again, we go for a third crop, intensive agricultural systems. Yeah.
So I qualified the criteria of questions.
Very well qualified.
Well qualified. You are recruiting new microbes and they are fighting with the indigenous microflora already. Allochthonous. That is the allochthonous microbial introductions with the indigenous one.
Right.
So it would be a novel approach to pursue further. So I would request Ayushi, please go ahead.
Okay. Thank you so much.
Thank you, sir, for your enlightening presentation. Now, we would like to request Dr. D.K. Chaudhary to felicitate Dr. Birinchi Kumar.
We are very fortunate to have you with us. It's your first visit in Amity, and you met with Professor Verma also, right? So I would request all the colleague, please come forth and join me for felicitation, please. Dr. Amit, Dr. Naveen, Verma sir, Anukul, you may join. HP, please come up. Swati, Menka, Monica, Jagrati. Surbi also, and Aarti and Shalini and Smita. Come forth, please. It is an opportunity for us, right? Please.
Now, everyone is requested to kindly proceed for lunch at E2 foyer. After that, kindly join back for the next talk by Dr. H.B. Singh in the same hall. Thank you.
Yeah, we can have a chat on, tea workshop. शर्मा जी ने तो जमाया बहुत जबरदस्त हुई. I was
शर्मा साहब तो पूरा जमा दिए पूरा.
I was so happy to see his presentation. Wonderful.
Now, we would like to invite Dr. H.B. Singh for his talk on registration of microbial biopesticides for commercialization. He is currently working as Distinguished Professor, Department of Biotechnology, GLE, University, Mathura. Prior to his current assignment, he also served as professor at the Department of Mycology and Plant Pathology, BHU. He has been honored with 22 national and international prestigious awards, including CSR Award for S&T Innovation for Rural Development of CSR, New Delhi, in recognition of excellent work on plant growth promoting microbial bioinoculant for enhanced crop productivity by former Prime Minister of India. CSR Award, Lifetime Achievement Award for Herbal Crop Protection by VS Research Foundation, Award of Honor of Excellence, ISMPP, and many more. He has guided 24 PhD students, 3 postdoctoral, and 2 women scientists, and 23 MSc Agriculture students. He successfully completed 21 research projects funded by various funding agencies such as DST, DBT, ICR, and many more. He published several research articles and review papers in highly reputed international journals and international and national journals. A very warm welcome to you, sir.
Thank you for a very nice introduction. I'm sorry, in fact, I was supposed to deliver talk in the forenoon, but because of the pre-engagement, I requested the organizer to postpone my talk. And it's one way it is good, because you have heard an excellent lecture of Professor V.K. Sharma. Now you have fair idea about what the microbiome is and what way the scientists can really help in mitigating the climate change and other things. Friends, microbes are very close to my heart, and last 35 years, I'm concentrating my research work on microbial pesticides. There are different kinds of pesticides. And whenever I used to deliver a talk, P. Pharmospora indica discovered by Professor Ajit Verma, always, take place in my lecture. Why? Because he is the one who introduced this microbe for the first time in India, and a lot of research work has taken place. And you have heard the eminent speaker in the morning. So I will be, in fact, talking only on a regulatory requirement of microbials. But what are the regulatory requirements? Just a minute, let me share the screen. I have some problem in my computer key. That is why I've sent my.
Shall I share from here only?
Yeah, I think that would be better, dear. Because I have some problem in my computer key. I was you share from your side. That would be better. Yeah, fine. You can move to the next slide. Yeah. So why registration? This is a big question asked by academia particularly, and many of my friends in industry also. They always talk that the registration part has to be removed. If not removed, then there should be relaxation in the registration. I don't agree to either academia or the industry. Government with well thought of idea introduce the registration with the aim that good quality biopesticides or bioelectronic may be provided to the farmers. And that was the reason through the Act of Parliament, the Gazette was notified on 22nd March 99, keeping 9 microbes initially in mind. For the student, particularly the young faculty sitting in the hall, I just want to tell you that the idea putting under the Gazette notification and putting under regulation of biopesticide was only to provide the good quality biopesticide. What was happening in the country prior to that, that most of the companies, they were producing low quality biopesticides. They were so spurious, no microbial counts were there. And many businessmen, what they were doing, they were producing quick money and disappearing from the market. And that erodes the confidence of the farmer in biopesticides or biostimulant or biofertilizer, whatever you may call. There are so many terminologies. So for the students, I want to clarify here. Biopesticide are 3 different kinds. I'm talking only for microbial, not for plant, and the plant biopesticide means the botanical. Then biofertilizer, you all know, a lot of discussion has taken place in the forenoon by Dr. Chaudhary and then Dr. Sharma. And the new legislation has come in the country. Is last year, the again notification has come on biostimulant. And a lot of hue and cry was there in the country for regulating this product because, you know, in the market, so many biostimulants were there, but they were not regulated. And that was the reason government thought, let us regulate biostimulant too. I'm not covering the biostimulant in my talk because the guideline has already been finalized. Gazette notification is already published, but till date, it has not been implemented. And therefore, those who are interested to know much about biostimulant guideline, they can surf the web and they will get the information. So 108-page document, it is available on the website. Okay. So now that was the reason, in fact, for putting biopesticide under regulation, and regulatory body, Central Insecticide Board, CIB, RC, Registration Committee, located in Faridabad, very close to your place, very close to your place. So they are the regulator in the country. So meaning thereby any scientist or any company or any manufacturer want to produce biopesticide based on micro or on plant products or their metabolite, they have to register their product with Central Insecticide Board, RC. Next one, please. Next slide. Yeah. So as I was telling you that the first Gazette was published on March 26, 99, and initially 9 microbes were put in the Gazette schedule. On the left panel, you can see antagonistic fungi and bacteria. They are Bacillus subtilis, Pseudomonas fluorescens, Glycolidium species, and Trichoderma species. And on the right panel, you will see entomogenous fungi and viruses. They are Weberia avesiana, Metarrhizium anisophili, Verticillium lecani, granulosis viruses, and nuclear pyrrhotic viruses. These were the 9 micro put in the Gazette of India. But later on, next one, please. Means till to date, I can say 25 more microbes they have been added in the Gazette schedule. And the last one was Piriformospora indica, which was discovered by Professor Ajit Verma. In the morning, you know, the Dr. Amit was mentioning about mycorrhiza. So I think we should not call this as a mycorrhiza. Let's call it as a endophytic microbe. because mycorrhiza is slightly different from the one which piriformis indica. So now you see here the piriformis indica has been put in the gadget as a biopesticide, meaning thereby if you want to register the product of this microbe under biopesticide category, you have to follow all the guidelines set by the CIBRC. But if you want to register this product as a biofertilizer or as a biostimulant, you can go to fertilizer control order. That is little liberal, and data required for registering a product under biofertilizer category is little less. And therefore, label claim, if you are putting on your label claim Piliformis indica as a biopesticide, meaning thereby they are controlling certain diseases, then you have to do that exercise with CIBRC. And perhaps that was the reason Professor Verma thought, let us talk something about root tonic or root whatever may be, because that way you can escape the process of registration under CIBRC for biopesticides. So these are the microbes listed. This is not the final list. See, friends, those who are having, I was listening the inaugural lecture. Many more microbes, they have been discovered, and the work is going on in MIT School of Biotechnology. Now, if they have publication on those microbes, they feel that the microbe has a potential activity for controlling various diseases, they may send their request to the CIBRC for inclusion in Gazette of India. So there is a subcommittee under the CIBRC. They will examine your case. They will see whether the publications are index journal. And if so, if publications are there, patents are there, then they will include that in the Gazette of India. So notification will come, and that is the reason every year you get notification containing one or the more microbe. So my humble submission to MIT guys particularly, if they have discovered some microbe having the potential of biocontrol activity, they can write to CIBRC for inclusion in the schedule. We call it in schedule. Once you write that my microbe must be put in the schedule, then they will approve it through the subcommittee, and then it will be sent to the Government of India for publication in Gazette of India. Okay, next slide, please. If you see the scenario in India, I think we are the biggest so far as the number of industries are concerned in our country. Total number of 970 microbial products are developed, highest in the world. 970 products, but I'm extremely sorry to let you know that 90% product manufactured by Indian companies, they are spurious. I don't have any hesitation in telling. I have spoken this type of thing in front of Honorable DG ICR and many more big shots who matters in agriculture. So 90% products are spurious, and that is the reason farmers, they do not have much interest in using biopesticide. Now, we as a scientist work like Professor Ajit Verma spent his lifetime on microbe, and he discovered the microbe, from the result having a unique property, but it took 25 years to commercialize that particular. Still, the product is not very popular among the farming community, though the microbe is very, very powerful. I can say it's a really magical, having a magical property. The reason is once you, as a scientist, transfer the technology to industry, then industry guys, they do not have proper methodology to keep your microbe for a prolonged period. I have visited so many industries in the country, and many industries, what they do today, they produce formulation based on talc powder. You know talc powder? In foreign countries, in Western countries, talc powder is not at all used for any formulation today. But still we are using the most old technology, I can say. Now, how to improve this? There are a number of things. Those working in microbial formulation, we will discuss in detail. Because what kind of things are to be added in the product so that you can prolong the shelf life of microbe. Number one, this is one. Number two, UV protectants. They are very, very essential, because once UV protectants are not added in your formulation, then once they are applied in the field, the way they are supposed to work in the field, they don't work. And that is one reason why the microbes, they fail in the field once they are applied for controlling a particular disease. Now, a lot of things has to be done in formulation technology. I don't find anybody in the country who is specialized in formulation technology. We have a National Institute on pesticide formulation technology. The whole government machinery is talking nowadays to replace pesticide with biopesticide, biofertilizer, and biostimulant. But we do not have an National Institute which deals with the formulation technology. And therefore, I firmly believe that we require institute, dedicated institute who can work on microbial formulation technology. Now, if you segregate the products into different kind of registration, see, in the CIVRC, there are two distinct type of registration. One is the provisional registration that is given for 2 years to a company or an individual. That is known as under section 93B. And this is only for 2 years. At the most, on the request of the company or scientist, they give extension for 1 year. Meaning thereby under 93B, you will get license for only 3 years. Beyond that, license expire and correct manufacture that. But the other category is known as, regular license, that is known as 93B, 93, section 93. So if you see here, 791 products are registered in provisional category. And most of them, they are fly overnight companies because they make the product for 3 years in the name of a company, again change the name of the company, take 93B, after 3 years again they change the company. This is what was happening in CIV, and we deliberated at depth and decided, let us put a bar on granting 93B. So now CIV has decided not to give provisional license to anybody. Notification has not come, but I had a talk with ADG, and in principle it is happening. CIV is not granting 93B to any company now. So now what will happen? You have to submit the complete data to CIVRC, and you will get a regular license under section 93. And under section 93, today we have 68 products. And out of 68, you will get 43 are fungal-based product and 25 are bacterial-based product. Next one, please. Our group, wherever I was working, either at CMAP or NBRI or Banaras Sundar University or even at GLA, we focus on trichoderma. Siddharth, my domain, and I work a lot on trichoderma. And you see here, Indian market, we have 355 products based on. So I say trichoderma is a hero of microbial pesticide. Okay. Now, under this category, only 30 products are registered. You see here, they are under regular license, and this one is under provisional. 325 products are under provisional registration. As I said in the beginning, the CIV has decided not to give any more provisional registration, and therefore now anyone interested in getting license, they have to go for 93, that is a regular license. Next one, please. Next, please. Yeah. Now see the country's infrastructure for production of biopesticide. This is the data available on the website of Government of India, Ministry of Agriculture and Farmer Bureau. We have 360 biocontrol laboratories in the country. They do not have those which are run by private players. Only private sector laboratories are there, which are funded by the Government of India. Meaning thereby there are about 300 plus industries manufacturing biopesticide in our country. So 300 plus 361 means about 650 plus industries are manufacturing biopesticide in our country. Out of that, you can see here, private sector laboratories are 141. 98 labs are state biocontrol laboratories. 49 labs are run by ICR Institute or State Agriculture University or DVT Institute. 38 numbers private sector laboratory, they are government aided one. And Government of India Central and Pest Management Centers, we have 35 in almost all the state of our country. But you will be surprised to see, next one please, the production which is happening in our, next slide please. See again, this is the latest data available on the ministry's website. We are able to produce only 8,898.92 metric ton of biopesticide in this country. This is just a meager, I can say, for controlling diseases using biopesticide. So just talking that the chemical pesticide should be slowly replaced with biopesticide, in meaning thereby you have to increase the infrastructural facility of our country so that the production of biopesticide is at least 100 times what is produced today. And then only we can meet the requirement of farming community. Okay. Earlier data, you see 7,000, approx 7,000 to 8,000 metric ton we are producing, which is too less to be used by the Indian farmer. And if the government is underlining that we should produce more organic, then I think we have to produce many more. And we need more factories, we need more industries, and we need more research institutions and universities to produce biopesticide for the farming community. Next one, please. Now, friends, as I told you, there are different type of registrations set by the CIVRC. I cited provisional registration under section 93B. It is given 2 years first time. Now totally stopped. You will not get. I had a talk with Honorable ADG Plant Protection Biosafety. He said we are not granting any 93B. Now the other is regular registration subject to submission of complete data set under section 93. That is for lifetime. Once you apply, get the regular license, until you produce the good quality biopesticide, your license will be valid. But if your quality is bad, the regulatory body has every right to cancel your license, and you will be punished also for that. And there is another provision in the law, repeat registration. Me too. After regular or original registration, any person can apply for registration under section 9(4). This is a very good thing which has happened in case of biopesticide. Earlier it was not there. Meaning thereby that if somebody has registered a strain with CIVRC, and if some other company or individual wants the license, you can use the same data and get the license without developing any sort of new data. The only thing you require a certificate from the inventor that we are transferring this data to this individual. It may be individual, maybe a company, and you will get direct registration under 9(3)B category from CIVRC. Now, in the morning session, it was discussed that now consortia biopesticides are also included in the schedule. I'm extremely happy to share I was one of the core committee member of the CIVRC when the guideline was supposed to be framed. And I, as a scientist, produced about 30 research papers and several patents to CIVRC that microbial consortia must be included in the CIVRC. It took 9 years to get it done, and I'm extremely happy. I congratulated Honorable ADG Dr. Dubey that we tried for the last 9 years, but you succeeded. Done. Now microbial consortia has already been notified. I will be showing you on which particular date it has been notified. But till date, no microbial consortial product have been registered with CIVRC. So those who are working on microbial consortia, I think Dr. Sharma, my colleague from Banaras University, with all humility, I can say we are the leader in the country so far as the microbial consortia work is there. We have published about 24 papers and few patents on microbial consortia. And hopefully in future, we will be again able to transfer this technology to some good industry for commercial production of biopesticide. Next one, please. Now, in the beginning, I said why the legislation was put forth for registering the biopesticide based on microbe. Only concern of the scientist and the government was quality control. So what is quality control? There are two different kinds of quality control. One is production control, the other is process control, and that is the internal quality control. So if you are working in a university system, developing a technology, so what you have to do, that you have to develop a protocol for production control. Not in a tube or a small flask, you develop a technology and think that big industries will come and take my technology. No, they will never come to you. You have to produce on a big way, maybe fermenter or bioreactor. Now, these things are all available in MIT. Not only in MIT, most of the universities in the country, they have fermenter and bioreactor. You are close to Delhi. IIT Delhi has a big fermenter. Many private companies are going and getting their microbe cultivated there and then formulating the product coming back to the industry. So if anybody want to have a startup on biopesticide, biofertilizer and biopesticide, no need of buying a fermenter or bioreactor. There are institution, I told you, like IIT Delhi, several IITs, several central universities, including MIT, they have a fermenter. So you can use the fermenter and bioreactor for internal production control and then process control. You have to learn. See, the microbials are very, very tricky. It's not that you grow anywhere the way you want. There are specific media, there are specific way. There are methods of solid state fermentation technology, liquid state fermentation technology. And so every process has to be understood thoroughly by the scientist, particularly the student or whosoever is doing this. Unfortunate part in our country is most of the industry, I can say 99% of the industry, they do not have technical manpower for running those fermenter and bioreactor. If you go to an industry, they are telling, we have hired a BSc AG or we have a science graduate. What they will do? They cannot do that. So you need a specialist of fermentation technology who can run a fermenter. You see the fermenter capacity, the industries, they are using 10,000 liter or 1 lakh liter. So if somebody is using 1 lakh liter fermenter, imagine if 1 lot is contaminated, he is going to incur loss of several lakhs of rupees. And therefore, I humbly request my industry friends, if they are having in this type of facility, they must hire technical trained microbiologists who can properly handle the fermenter or bioreactor. Then another is product control. That is the job of the regulator. That is the CIBRC. What they do, they have fixed certain guideline that if you have made a product, the product should look like this. It should have this much pH. It should have this much CFU. It should have this much what. I will be telling you all those things in my future further. Next one, please. Next slide. Yeah. Now you see here, product control refers to the quality of the final product that leaves the factory and which need to perform according to registration and customer satisfaction requirement. What is happening most of the time, industry guys fight with the regulator that at the factory side, we produce the best quality biopesticide. But while in the transportation, suppose you are producing biopesticide at Noida and transporting from Noida to Kerala, or from Noida to Srinagar, or from Noida to Northeast, Nagaland, Assam, Tripura, Meghalaya, maybe. So if it goes in truck or in railways, it will take 10 to 15 days. Now the biopesticide will be packed in primary and secondary and tertiary packets. Not that you are simply packing in a gunny bag. It should be packed properly. There are strict guidelines for packing material also. So just scientifically, you think during the summer, the temperature in India touches somewhere in between 45 to 48 degrees. So in transit, in the truck, the temperature is about 55 degrees. So in 55 degree temperature, the truck is moving from Noida to Kerala, from Noida to Northeast, and from Noida to Srinagar. So what will happen? Whatever little microbes they were there, at the time of preparation of the biopesticide, they are completely killed. And that is the reason 90% biopesticide, they do not work in the farmer's field. So I believe as a scientist, we all have to work hard how to increase the shelf life of the product. This is very important. Otherwise, you have to develop a cold chain of transportation of biopesticide. Otherwise, we all will fail. We will fail and we will not have any response from farming community. Then the product must meet the product specifications set by manufacturer until the end of claimed shelf life. What is happening? If you have a commercial product, must have seen by now, then you get that shelf life is 1 year or 1.5 year or 2 years. Some manufacturers are even writing 3 years in a talc-based formulation. As a scientist, I don't believe, I have never seen any product which survive at room temperature for 3 years. Tell me, how can a microbe which grows only in 25-30 survive at 50 degree in a talc-based formulation for 3 years? That is a big question mark. It has to be asked by the scientists who are claiming that, yes, I have a product having 3 years shelf life, with due respect to all of them. Quality control must ensure that the end user receive high quality product. That is the motto of the government and motto of the scientist also. We all feel that my farmers, my user of biopesticide, they should get a good quality. Now the biopesticide company must ensure that the whole chain is well aware of quality issue and that those involved must act accordingly. I will tell you my own experience, friends. Many times I go to dealer and distributor and ask, I want this much biopesticide. Can you provide me? 90% time you will say they don't have. Why the dealer distributor, they are not keen in selling the biopesticide? One, that poor quality shelf life. And the other is that the farmers, they don't come for this. If at all they come, they tell, okay, I don't have, but I have a product. You take this, this will take care of your crop. And what they used to give earlier, that was a enzyme-based product, and we were calling it a zyme. So farmers are very well aware that what is zyme? Zyme is plant growth promoter. So it may be a plant growth promoter, it may be a enzyme-based formulation, it may be humic acid-based formulation. There are n number of formulations available in the market having enzymes. So those enzyme-based formulation, when the farmer carry, he spray on the crop, they get a very good response because the enzymes act very quickly, but they do not control disease at all. They again come back to the dealer and ask that the disease has not, so then they will give the pesticide. So hardly you will get any dedicated shop in the country that they are selling biopesticide. I recommended 2-3 times to CIVRC that please make sure that there are specific shops which are selling only biopesticide and biofertilizer and biostim. But till date, it has not happened. So till you promote this type of thing, particularly and make it compulsory that if he agri graduate there, only he will get this license for selling the biofertilizer and biopesticide. But these are all policy issues, and small people like me cannot, enforce the government to do so. But it will. See, I was listening beautiful talk of Dr. Sharma. So nice. You see the in vitro, then part trial in greenhouse, they are doing wonders. But if they are so wonderful, why my farmers are not buying those products? This is the reason which unfortunately many of the scientists, they don't disclose. But I feel it must be told to the young generation, particularly the young budding scientists, very talented scientists, in MIT. I have personally contacted many times, I have visited MIT campus several times, not the Noida one, of course, other campuses. Next one, please. Now, listen to me very, very carefully. Now I will be telling you what are the parameters set by the Central Insecticide Board Registration Committee. Now, one thing they have said in the law, that number of infective propagule, we call it colony forming unit. They have fixed this one, that this should be the colony forming unit in the formulated product. I will tell you how much colony should be there in 1 gram of product or 1 ml of product. Then moisture content. If it is a talc-based formulation, then moisture content guideline also has been fixed by the CIVRC. And the third and most important component is microbial contamination. So they have listed only 3 micro that they should not be present in your formulation. If at all they are present, other microbes, their population should not increase more than 1 into 10 to the power 4. But in regulation, nowhere in it is mentioned, identity check is not part of the product control. So as I said, I was made, the advisor in the CIVRC for 3 years for a special committee. So I recommended several times, but it never happened. I want from this forum, and I request Dr. Chaudhary that in recommendation, please keep these points so that in future you are close to Faridabad, so you can even pursue with CIVRC that if you really want to have a good quality biocontrol, you have to enforce this guideline. And till you are not doing this, poor quality products will be sold in the market. So identity check must be a part of the product control. That's what I want. Next one, please. Now in the CFU count, friends, you know what has been mentioned, that the CFU should be 2 into 10 to the power 6 spore per ml or per gram on selected media. It means you buy a packet from the market, see on the selective media whether 1 gram or 1 ml of the product is carrying 2 into 10 to the power 6 spores or not. If it is 10 to the power 6, it means your product is at par with the guidelines set by the CIVRC. This is what they have said. Another thing is a product should not have less than 1 year shelf life. Otherwise, the dealer and distributor, they will not keep in their shelf. And that is the reason almost all Indian products available, they are showing 1 year, 2 year, and 3 year shelf life. Truth is something different. Another thing which CIV is telling, the pathogenic contaminants such as Salmonella, Shigella, and Vibrio should not be present. They should not be present. Other microbial contaminants not exceed 1 into 10 to the power 4. But what industry does, I will tell you, they feel other way. If the Salmonella, Shigella, Vibrio is there, they say it is under the control of 1 into 10 to the power 4. It should not be there at all. Other than these 3 human pathogens, other microbial contaminants are allowed, but their limit also should not exceed more than 1 into 10 to the power 4. Now see here, which was my recommendation, which has not been accepted till date. That is why I have put a red color here. Only for MIT guys and for Dr. Chaudhary personally. No regulation on harmful toxin produced by human pathogen. Hence, each batch testing may be mandatory. If as a scientist we have put our head for 35 long year or 40 year and learn little bit of microbe, why not, tell the regulatory body that these are essential component of biopesticide and they should be put, and then only you can regulate properly. So these are the, you see, aflatoxin producing aspergilli. There was a product developed at IRI. I will tell you the story of that product. Professor Vinita Sen developed that product, and that was named as a Kali Sena. Kali Sena or Kali Sipahi, 2 products. They were licensed to Cadila, and they were used by the farmers for controlling many soil-borne diseases. And it was claimed that this particular fungus does not produce any toxin. But what happened? When the company took technology, started producing, and the pesticide was sold in market, the Gujarat farmer started complaining that their groundnut is getting infected by aspergilli. So a high-level committee was set up, and then it was found that the biopesticide, which was produced by Cadila, is producing toxic compound, and that is the reason the crop is infected by aspergilli. See, they mix the strain. They lost the original strain developed by Dr. Vinita Sen. She spent 10-15 years on developing that. And she retired long back. Now, unfortunate part again in this country, you develop something, you retire, your culture goes with you. You don't have a system that if you retire, you have to deposit your culture to the institution. You have to deposit your culture to national repository, international repository, because these are the national assets. So that non-toxic strain developed by Vinita Sen through a product of biotechnology has been lost. And today, the product has been banned in the country that you cannot make that product using the same strain. So this is what is happening, and that is why I feel the harmful toxin produced by human pathogen must be included in the next one, please. Now, again, this is an issue which I have deliberated at very at length at many fora. See, the maximum moisture content in the product should not exceed 8% for the dry formulation of fungi and 12% for bacteria. Meaning thereby, if you are making biopesticide based on fungi, moisture content should be 8% fixed, and for bacteria 12%. But our study, and not only our study, studies from all across the world, if you see, bacteria survival is higher at higher moisture content. So that also require revision or relook. So those working on bacterial and fungal formulation, please try to see and develop data how much optimum moisture content is needed for keeping that particular microbe for a prolonged period. So this guideline was prepared in 1999. That time data was not available till date. We have enough data. So we can suggest the CIVRC that please increase the moisture content in the formulated product so that the shelf life can be prolonged. So this again a recommendation, red one, can go from MIT side to the CIVRC or ICR so that it is implemented in future. Next one, please. Now, bioefficacy data also can be generated by the institutes I have listed here. Not everybody can do that. Only ICR Institute, State Agricultural University, CSIR Institute, and ICMR Institute. We requested CIVRC to include central universities having agricultural college. That was my initiation, I can say, long back in 2002. 2002, we initiated this process. Because in the country, we have central universities having agriculture institute like Banaras Hindu University, Aligarh Muslim University, and many more. Some new central agriculture universities have come up in the country. Now, as per the new guideline, you have to say that central universities having agriculture faculty and central agriculture university should also be included in the schedule. So, of course, through a gazette notification, central university having agriculture facility has been included, but central agriculture universities are still not there in the gazette notification. So I need a recommendation must go that central universities, central agriculture universities, like, we have one in Imphal, one in Jhansi, one in other places. There are in Bihar. So those can also be included so the scientists working in those institutions can develop data. My feeling is why to ignore the university like, JNU or Delhi University or many state universities are also having a wonderful faculty and wonderful staff, including some private universities. So if a recommendation goes, why not MIT University should be authorized to develop data for the bioefficacy? What is the harm in so? So legislation was passed long back, and I think we as a scientists have to think that some new organization they have to be added. And I think this is the right forum through which we can recommend the authorities to include private universities, central agriculture universities, and other institutions which are not included in the bioefficacy trials. Next one, please. The biggest hurdle for a scientist in developing technology is toxicological data. And my friend Dr. Sharma always talks that from where to get money. He has a very good micro, particularly, the rhizobia. Of course, they also require now under the new regulation tox data. And then, of course, Pseudomonas, other bacterial microbes he had having an excellent property to control disease. So this is the toxicological data requirement. And there are many toxicological laboratories in the country run by private and government. But But one thing was added in the schedule that only GLP accredited lab can generate tox data. One good thing has happened again in the CIVRC, that they have removed the word GLP accredited lab, meaning thereby non-GLP labs too can generate tox data. So this is a good news for all of us. The notification already come. It has been placed on the website of CIVRC. So you can get the tox data generated by non-GLP lab also. It will be cheaper. But in general, the tox data generation cost is very high, and that is the biggest hindrance for a scientist to develop technology which can be commercialized. And in all these things are required, as I know, tox data person, you will understand that these are the tests required by the labs, not by you. Next one, please. Then packaging and labeling is another important thing which is very, very essential for commercializing any technology. We have only one institute in our country for packaging, that is Indian Institute of Packaging Industry, located in Mumbai, and they have a branch office in Delhi also. So close to Delhi you are, so I will tell you the address. Palpalganj is the place where the office of Indian Packaging Industry is there. That is run by Government of India. And fortunately, the director of that institute is from our university, that is from Banaras University. Friend of all. So he is there. So anyone can go and approach him. Mr. Tanveer Alam, Dr. Tanveer Alam, he is the director now. Very dynamic fellow and very helping. So you can go and see what kind of packing materials are available for all kind of things, including medicines, food items, and so on and so forth. So the data will be developed by them only. Nobody else can develop data for packaging and labeling. So they will develop data for you. They charge nominal money. So this is one requirement for registering any product with CIVRC, that packaging and labeling information has to be generated by the institute. Next one, please. Now, there are some recent amendments. I will talk on that. Next one, please. Now, again, when Dr. Chakravarti was ADG, in that committee, we decided that how to catch hold these fellows who are making spurious pesticides, biopesticides. So Dr. Chakravarti is a biotechnologist, and I worked on trichoderma and other biocontrol agent. So we decided that let us put a DNA barcode compulsory for all the products available in the market so that we can catch hold. So on the initiation of the then ADG that time, Dr. P.K. Chakravarti, this particular thing was passed, and then DNA barcoding was made mandatory for all the industry. And for that, a dedicated institute, ICRNBMO, National Bureau of Agricultural Important Microbe, has been empowered to develop the DNA barcode data. It means anyone developing technology, you have to submit your culture to NBMO for barcoding. They charge money for that. So that receipt has to be kept with you very, very, thoroughly. Otherwise, you will lose the license, because that receipt has to be submitted to CIVRC. And then technical audit by expert. This also was recommended, but not approved by Government of India. So this part has not been implemented till date because it has not been notified. We wanted that technical person like scientists or the PhD scholar who are trained in microbiology, they can inspect the industry and say whether the process is true or not, whether the microbe which they have taken from the per scientist is same or not. All those things we said, but it has not been approved by the government. But yes, culture deposition has become mandatory now for barcoding, and with the barcode you can catch hold that the technology taken from this particular institute, the same microbe is going along with the product. Next one, please. Now, this was the new thing which happened in CIVRC, and people are so happy with Microbial Consortia. So this notice was published on May 23rd, 2022. And there they have said, I have just quoted the same wording. The registration committee in 439th meeting held on 25th April 22 has approved the guideline of biopesticides along with consortia of biopesticide under Insecticide Act 1968. But the biggest problem with this regulation is there are companies in India already manufacturing biopesticide having 10, 15, 20, 50 combinations. Now, how to take out the CFU of 50 different microbes, because that is the only criteria set by CIBRC. And that is why another committee has been constituted in CIBRC, headed by Professor A.N. Mukhopadhyay. And there are 2 more scientists with that committee to decide how to calculate the CFU of different microbes which are added in consortia. So rule is there. CIBRC will register consortia in future, but till date, not a single product is registered with CIBRC. So still CIB has to do a lot of work, homework for registering a consortia under the Biopesticide Act. Next one, please. Previous one, please. Yeah. This is what I wanted to share now from IPR to commerce. Because earlier people were developing technology without protecting IP. I can say our group was the pioneer in the country to patent first trichoderma from India. And we patented in 99, developed technology and transferred to a company known as GSFC. Here you can see Gujarat State Fertilizer Chemicals. It's a very big company known for producing urea and DAP. And here you can see the logo of my previous institute where I was working, NBRI Lucknow. And we charge 1 crore as a license fee for transferring of this technology. So a teeny micro technology was transferred way back in 2002 by charging 1 CR from the company, and 40% of the total money goes to the institution, 60% goes to the institution, and 60% is distributed to the inventors. I was listening very carefully in the morning session. Dr. Silva Murthy is a wonderful orator. I worked with him in a DST task force for 3 years. He really is a wonderful personality. Since he's in MIT, you all know, he was earlier the Director General of the DRDO, and after his retirement, he joined MIT. So I worked with him. So last 3 years, I could understand about MIT and about him. So very nicely, he narrated the whole story about the futuristic model, how the MIT should look like, and how the Pilifarmos indica product in the future. So what I'm trying to tell you, this particular technology was sold by taking 1 CR way back in 2002. That time ICR was selling technology in 75,000. Now this is you who have to decide how to sell technology and what should be the cost of that. Even today, I feel very sorry, and I always talk to directors of the institute and then DG also, that sir, the rates given by your department is too less. What is 1 lakh or 2 lakh today? A professor gets 3.5 to 4 lakh salary per month, and he developed a technology in 4 to 5 years involving so many research scholars, so many technicians, lot of infrastructure of the university use or institution use, see their electricity bill and so on and so forth. I do not know why the people are keeping 75,000 rupees. Anyway, that is a different story. So this was the first product launched by GSFC Barodara. And that time our Honorable Prime Minister was the Chief Minister of Gujarat. I'm very happy to share that he is the one who launched our product. In 2004, it was launched by him. Next one, please. The same technology was, no, go back. There are a few slides. Go back. Yeah. The same technology was transferred to Balaji Crop Care Private Limited, Hyderabad. Next one, please. We license the same technology to government of Uttar Pradesh. Next slide, please. This is another product developed by our group, my colleague Dr. C.S. Notia and myself, Bacillus subtilis. This is the only product based on Bacillus subtilis in India, and therefore we are the topmost seller. Means the company used to sell approx 50 crore plus this product in the country because there is no competitor. We don't have any product based on Bacillus. So Bacillus thuringiensis we have, but we don't have Bacillus subtilis. So here again, you will find CSIR logo on the top and technology transfer and so on. Next one, please. Now, as a scientist or as a research scholar, you always feel that my news must go to the newspaper. You feel very happy. I was so happy that time when my news was published in the CSIR newsletter. Next one. And the technology transfer was done by none less than the Director General of ICRD, then Dr. R.A. Maselkar, very familiar name you can see here. He is the Director of NBRI that time, Dr. P. Puspangadan, and MD of GSFC Barodara and Technology Transfer Head. Now see the importance. The scientists who spend lifetime in developing technology is not in the photograph. So that is the Indian culture. Somebody does the work and somebody take the the press coverage. I don't have any hesitation now in sharing this. I'm a retired guy, I can tell. I was willing that time that I should also be there on the dais. But technology transfer had said, oh, no, DG is there and only 2 persons are allowed. Like if you are taking something in the hand of Prime Minister, then you have to take approval in the advance. So like SPG gives you approval, you can shake hand with PM or CM. So anyway, this is our technology. You can see here trichoderma-based technology has been transferred to GSFC Barodara. So only one picture I want to show you, because if you develop technology, I'm sure the founder of MIT, Dr. Chauhan, will come and transfer the technology and you will have a photograph with him. I think that will be a wonderful experience of a student or a scientist working in an institution. Next one, please. Go back. Again, happy to say that yes, we heard that the regulation part is very, very weak in our country. So we edited a book on agriculturally important microbes, commercialization and regulatory requirement in Asia. This book is available in soft copy. So anyone interested, you can request me or Dr. Sharma is there with you. He can transfer email to you immediately of this soft copy. Another book is on intellectual property issues in biotechnology, published from KV. They don't provide any soft copy. So anyone interested, they can buy the book from KV only. But of course, left one is available with us. And one good thing about these two publications are Anand Mohan Chakravarti, who patented first microbe on earth. You all know, he has contributed one chapter in both the volumes. So you will love to read the article written by Anand Mohan Chakravarti, that how he fought with Supreme Court of the United States for getting this claim that this genetically modified bacteria, there is human intervention and there is innovation, and therefore the patent has to be given on this. So friends, this is exactly what I wanted to share with you. And this program, really very happy to be part of that. In fact, I was supposed to come physically. I never visited Noida campus, and Professor Verma invited me several times. Whenever you come to Delhi, that was the only rider always kept with him. Whenever you come to Delhi, please give me a ring. I will arrange a visit for this campus. I couldn't do so far, but yes, of course, I have visited Jaipur campus. I have visited Gurgaon campus. I have visited even your campus in foreign country also. In Lucknow campus, I used to go sometime deliver a talk, but unfortunate part, I haven't visited Noida campus. I learned that is the most beautiful campus. Maybe in future, if I get any opportunity, I will certainly come and see. And then whatever little I know, I will be trying to discuss those issues with the student community particularly, because it's my habit if I go to any university for conducting MSc Viva or PhD Viva, I always deliver it on. Maybe on IPR or maybe biopesticide, biofertilizer, biostimulant, or how to write research paper or how to write projects, all these things, common lectures I used to give. So this is all, friends, I wanted to share with you. Now you can ask me any question pertaining to regulatory issues of microbes, and then how to commercialize that also I can discuss with you. I'm extremely happy to learn that MIT has filed more than 1,870 patents, and they have transferred few technologies. Also, I know this Piliformospora indica has been transferred to many companies. But if Professor Verma is there, I want to share something with him, or I can discuss this issue later on. Okay, thank you. Thank you, Dr. Chaudhary, for giving me opportunity to talk on this particular topic. I can just, these recommendations are there, but I don't want them.
Thank you very much. I am still here.
Sir, so nice to see
Go
you.
ahead. Please go ahead.
I will tell you, sir, one very secret, or a very, from this forum, I'm telling you, since the students are sitting there. See, number of mycorrhizal products are available in the market. So the biggest problem with them that the CFU count. So what they did, in fact, I checked in many formulation. They procured your culture from some sources and they mix with the VA mycorrhizal fungi. And perhaps nobody in India can identify whether it is a Piliformospora indica spore or it is a glomus or other mycorrhiza. So that type of, things are going on in the country. I think that also has to be stopped by you. And if you barcode your microbe and the companies are taking the license from you, you can devour those companies who are mixing Piliformospora indica with the VM population. These are my observation. I can tell you the name of the companies also are doing this. Because you are so generous, you have given culture to many people.
Okay, I've got a very basic question to you.
Sir,
How do you define mycorrhiza?
how we define mycorrhiza?
Yes. For my knowledge.
We have only read that the fungal association with the root. That is the most simple definition. A symbiont which lives in the root of plant is a mycorrhiza.
Yeah, that was all discovered almost 200 years ago.
Yes, sir. Yes, sir.
Right? And till date, no one could culture in the fermenter or in the batch culture.
Sir, I have seen a report. I have seen a report from in current science.
Yeah.
That glomus aggregatum is cultured.
I must say,
Yeah.
it was getting disastrous for the science.
Yes. Yes.
I
I fully agree, sir. I
was
fully
at
agree.
Max Institute,
Yeah.
and they sent a paper for Nature,
Yeah.
and then Barbara Mose was the reviewer of this paper, and she said it is not glomus, it's something else. And incidentally, I was in England. She told me, "It's okay, I'll go to Lucknow and find it out." When I went to Lucknow, they did not show me the culture.
Yes.
Some excuse आता है, आज वो नहीं आया, 3 दिन मैं वेट करता रहा वहां पर, मुझे
कभी नहीं मिला.
finally what happened, he sent the paper to Current Science, of which one of the scientists, very well-known scientist of Delhi University, was the editor-in-chief. He published that paper with a lot of colored photographs. And finally, when I disputed, I must take discredit for this one. I told, and the scientists were dismissed from CSIR.
Yes,
so
you
that
are
is
right.
a very novel example where a scientist made a wrong claim, duly authenticated by very senior scientists from Delhi University, and finally scientists got dismissed. Okay,
Right, sir, yeah.
so you know
That is a big story. Yeah. So I
Now-
think it should be a lesson to students also that nobody
they
should
must
dare
know.
to report.
Students must also know
Yeah,
that what a paper are not correct.
yeah, yeah.
Some of them are very wrong.
But sir, I'm so happy that this fungus, I think more than 200 research paper from across the globe, several
True.
patent books.
Very
I
true,
think,
very
yeah,
true.
yeah.
And by now 39 PhDs. Okay,
Oh,
now
great. Yeah,
my question is the following.
sir.
Why not I call P. indica as mycorrhiza? And you dispute it.
Sir, I was talking in many seminar and symposia about this microbe. I think my lecture never ends without talking about this particular microbe, because that was the last microbe I did in Gajan. So a very senior scientist objected, this is not a mycorrhiza, it is mycorrhiza-like fungus. Or it is endophyte. So I never worked on mycorrhiza, sir. Therefore, I am not the authority to, but I think it's safer to call endophyte.
Yeah, what happened, there are lot of, there are two things which you mentioned in talk, that why mycorrhiza could not become popular. Why endophyte could not become, because there's a lot of dispute from the industry. They want to promote the chemical fertilizer over the microbial fertilizer. A, B, there's also a lot of professional jealousy.
That's right.
I know I don't take the name now. And what they want, because they are very jealous of this.
Absolutely right.
This
Absolutely.
That's the one reason. Okay, thank
But
you.
sir, the biostimulant, one thing has happened in biostimulant guidelines, sir. The live microbes are not permitted in biostimulants. So this P. indica will only be registered as a biofertilizer.
And other last question I'll ask to ask you, what
Sir.
about carrier? What is wrong with talcum powder? Or
Talcum-
is there any better one which I can choose on?
No, I will tell you, sir. See, recently, sir, I took over as an advisor Syngenta Biologicals. I'll tell you, I interacted with the global team, the global head.
Hmm.
And when I interacted with him, then I thought we really don't know what is formulation. Now, what they are doing, sir, they are having a liquid formulation
Hmm.
with very high CFU,
Hmm.
and shelf life is really 2 years at room temperature. Then I asked the chemical, he said, you have to come to Switzerland for 15 days, learn this and go back to India and tell our guys there. I said, sir, I'm an old man, I will not come. Let my scientists should go. So I deputed one scientist from India, a lady scientist, and she has gone to Switzerland. She came back. Then I asked that lady, what you learned? She said, sir, they have a trade secret of that. So they have given me a powder-like thing. And once you add that, the shelf life is increased. Now, what I presume, that powder may contain some antioxidant compound and UV protectant. None of our industries are using UV protectant in biological preparation. But the Syngenta is using, not only Syngenta, Bayer is a big giant again in chemical, they have entered in biopesticide. Now these two major giants, they have entered in the country for production of biofertilizer and biopesticide, and they have biological technology of formulation. I wish somebody from India should come up with this one, that we have a liquid formulation having this much CFU and you require very little dose. Because I, sir, visited US in Dr. Gary Harman's lab, who is a father of trichoderma, and his recommendation is 5 to 10 milligram per acre of seed. I tried my level best to know, but he never allowed any Indian to enter that particular area where the work was going on. So my visiting as a scientist was a futile exercise. I couldn't learn. But as Indian, here if somebody comes, we open our heart and see Hanuman ban jate, ye dekho bhai, hamare paas ye hai. So I think my young generation must learn. They should also know we do not We should not tell any such thing to anybody. So liquid formulation is there, but nano formulations are also coming, sir. Now a lot of nano formulations are there. And in India also, some companies, Pratishtha and other companies, they have started producing nano fertilizers, and nano biopesticide also work is going on there. So maybe in future we will get nano formulations, and hopefully Dr. My friend Dr. K.R.K. Reddy is very, very, good in good microbiology, and he's the one who launched the first microbiome based product in the country, and he bagged several awards. So I think we have to learn something from industry also, that how to make good formulation. We are poor in that, sir. We are still poor. And one thing I forgot to tell you, if you plate talc powder, you will get all contamination of microbe, and therefore talc has to be sterilized. The Shigella, Vibrio, Salmonella, they are inherently present in talc powder.
Yeah, for your information, talc powder we sterilize 3 times with the gap of.
Yeah, then that's fine. But sir, for industry, it is not possible. I will tell you, see, the industries where we have transfer technology, we insisted, but they said we cannot do it because that is not practical, because there's a lot of energy is required for that.
Yeah.
And that becomes uneconomical for them.
What is the alternative, sir? What do you suggest?
Sir.
What do you suggest the alternative?
Alternative?
Yeah.
Liquid, sir. Liquid formulation, sir.
And liquid formulation, there is one problem, transportation.
No, there are very good packing available now, sir. That's why I'm telling you can send somebody to Delhi. Tanveer Alam is the director of, institute.
Okay.
You will get 1,000 different kind of packing material from across the world. You select what kind of packing material you want for your product.
Okay.
We will design for you, sir. Yeah.
Appreciate your suggestion.
Yeah, you can take the number from me. I will send you the number.
A very good book. I have made 128 books from Academic Press London and Spring of Allah. And he is one of the editors of the book. Therefore, I took liberty to talk to him very freely.
Thank you, sir. Thank you. Yeah. I feel honored to have a book with you, sir. You and Professor Manoharacharya, great person in my college.
Yeah, but Mathura is not far away.
Students.
You are in Mathura now. You are not far away. I think in the next some days, I'll invite you from Mathura to come to Delhi.
Maybe, sir, next time if I'm in Mathura, I will take Anukul and then come to your campus.
Thank you, sir. Thank you.
Yes, sir, I have a question. I am Dr.
Yes,
Anil.
yes, sir. Please feel free.
Sir, you spoke about Kali Sena, that is, I think AN27 strain.
Yeah.
It's banned now. You said that.
Not banned. See what happened.
Okay.
The strain has been lost by the company and nobody is there to supply the same strain to them. That was the reason they banned the product in Gujarat.
Okay, but it's still being sold, sir. It's a very high price, 2,800 to 3,200.
Sir, in India, you can do anything.
Okay.
In spite of the ban, the product is manufactured by other company and they are sold for composting, for control of wilt and other part.
Yes, it
But
is there in
on paper, it is banned in Gujarat only because of the production of toxin. This is the official figure of it.
Thank you, sir.
So it was nice interaction, Professor H.P. Singh, sir. And truly regulatory aspect we must revolve by deploying the microorganisms. What do they do in the ecosystem? And sir, at one place you defined that 1 into 10 to the power 4, CFU. Huh?
No, that 1 into 10 to the power 4, Dr. Chaudhary, is for the pathogenic contaminants.
Right.
Other than Salmonella, Shigella, and Vibrio. Otherwise, you require 2 into 10 to the power 6.
Okay.
That is the minimum CFU required for
Mm-hmm.
fungal and bacterial population.
But don't you
Most
think
of the companies, they do 2 into 10 to the power 8 or 10.
Don't you think this is quite much concentration if we talk about the propagule related with the bacterial species or the fungal one? It is too much, in my opinion.
Not too much. What happened, that's Chaudhary, I will tell you. Initially, they keep high population with the idea that in transportation, some population will go down. But that is not the reality. What is happening, even if you put 10 into 10 to the power 10, in the transportation, they lose their viability. And that is why I'm insisting the cold chain transportation of biopesticide is a must if you want to have a quality. Otherwise, what is happening? Industry said, we produce this, at factory side, this is my CFU. At the terminal point where the product was given, they were having, but in between, the shelf life has gone. So what to do? So they are saved.
Mhm. So based on, it's not a matter of contention now, based on consensus, we always agree with your talk and
Sir.
we would welcome in Noida. You missed this.
I will love to visit Noida. In fact, earlier date, I was ready because I discussed with Ankul that every time I go to, Mathura by car. So I said, we will go together and then.
If
come
you
back.
come to Mathura, please let me know. I shall be there to pick you by my car, right?
Great, great. Yeah, next time
Okay.
certainly I will do that. Yeah.
Thank you, sir. Thank you. So
Thank you,
nice of
sir.
you.
Thank
Any
you.
students, if you have any,
Yeah,
my colleague has.
Hello, sir.
mainly they will, yeah.
Hello.
Yes.
Sir, myself,
Your face must be there. Yeah, camera is there.
so
Yes,
myself,
sir.
Dr. Naveen Joshi, and it's quite informative talk. You talk a lot about biopesticides. So it's really great to listen to you. My question is quite hypothetical, actually. So what do you think? What is the, when we can stop using chemical pesticides? What is the future of those chemical pesticides, what we are using now? Can we stop completely?
No, I will tell you, in fact, in another presentation, I have some international national data done by the research company. They have predicted that in future, the use of biopesticide is going to increase and the annual growth rate of biopesticide will be approximately 15.8%. So this is a win-win situation for all of us. The biggest problem in our country is we do not have good manufacturer. So if we increase the quality of the product, certainly it will be used by the farmers. But we cannot totally stop chemical pesticide. But what we can do, there are safe pesticides. So let us use safe pesticide, biopesticide, biofertilizer, biostimulant. Then slowly we will move towards the safe and sustainable agriculture.
Yes, sir. If as you said, we cannot. We cannot stop using these chemical pesticides like that. So
Certainly.
there is one more strategy we can apply.
Yeah.
So we can use the GM crops, genetically modified crops, which can degrade these chemical pesticide effectively in the non-toxic one.
You see, I will again tell you, dear. See, when the first GMO was approved in this country,
that's
cotton, you know the story.
Yeah.
Initially, they said no, lot of protest by the farmer, and finally the politician has to agree. Now, what is happening to GM mustard? If a person spent 25 years, see, several crores of rupees was given to him as a research grant from DVT. But what is the outcome? Develop the variety of GM mustard, but cleared also by all the committees, but is still on hold. So we are very poor in disseminating the newer technology. Particularly, we are very particular about GM crops. And the present government, I do not know how far they are serious. They are not very serious about GM crops.
They have approved one recently, so maybe
that GM mustard is approved, but there are a lot of hindrances in that also.
Yeah, true.
If you talk to Dr. Pentel, he will tell you how much he has faced the music of these regulatory bodies.
Actually, we are working on this aspects in a meeting.
GM is one good alternative, but I think better than GM will be the CRISPR technology.
Yeah.
Number of CRISPR crops are available now all across the world. Let us have that technology in India, and many scientists are working in ICR system. So future will be CRISPR.
Yeah, sure.
You can avoid that. Yeah, you can avoid that.
Thank you, sir. Thank you very much.
Yes, sir. Thank you.
Thank you, sir. And Definitely non-transgenic approaches, CRISPR. It is under non-transgenic.
Yeah, yeah.
RNAi also, it may be.
RNI also a wonderful technology.
So we may move by deploying the microbial determinants and incorporation into the plant system. If my colleague, he is plant biotechnologist, Dr. Naveen, he is deploying the CRISPR technology over here in alleviation of the biotic and abiotic stresses with the rice crop, maybe. Yeah. So thank you, sir, for nice talk and enlighten us. So please join us next time in person.
Sure, sir, sure, Chaudhary Sam. Now you have changed a lot. I don't know. Yeah.
20 years back, I was something.
You look like a real professor now.
Thank you.
That time when I met you in DVT, you were like a student.
Experience.
Now you look like a professor.
Thank you, sir. So nice of you.
Yeah,
Please
thank you,
join us
thank
in
you.
person later. So if Dr. Amitwa is backside. Dr.
Yes, I'm here.
So
Yes,
please
I'm here.
show your face to all of us so we can.
Yeah.
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 So we are going to start with the next talk. Shall be taken by the Dr. Amitwa Rakshit. He is from BHU. He's a great scientist again, along with the series. So Ayushi or Gaurav, please introduce Dr. and then he shall carry on.
Now, we would like to invite Dr. Amitwa Rakshit for his talk on microbiome underpinning agriculture sustainability, success story from the fragile ecosystem of the Indo-Gangetic Plains. He is presently working as an associate professor at the Department of Soil and Agriculture Chemistry at the Institute of Agriculture Sciences, BHU. He has also visited several countries, including Europe, Africa, UAE, and Bangladesh, pertaining to his research work and presentation. He was awarded with the VAST Next Fellow, BioVision Next Fellow, Darwin Now Bursary, Young Achiever Award, and Best Teacher Award at UG and PG level and state level by Uttar Pradesh Academy of Agriculture Sciences. He is serving as Review College member of British Ecological Society, London since 2011, member of Global Forum on Food Security and Nutrition of FOA Rome, and Commission on Ecosystem Management of IUCN. He has published 125 research papers, 48 book chapters, 28 popular articles, 3 manuals, and co-authored 29 books. We welcome you, sir.
Thank you very much.
Okay, sir.
Okay, my voice is coming. It's
So
clear,
shall
right?
I upload your slide from here?
So at the outset, I must convey my sincere gratitude to Professor Varma, Ajit Varma, who was always a dream leader for us. And I always wanted to meet in person. But unfortunately, this time also, because of some very compressing schedule, as well as my health, so that didn't permit. Otherwise, it would have been a great gesture for me as well to meet in person Professor Varma, Professor Chaudhary for inviting me for this particular talk, and Anukul. I think Anukul also deserves appreciation for this connection. Well, before I start, it's just like something, when you have a cricket match and your consolidation has been very strong because 2 of your openers have batted for you, whether it is Gill or whether it is, Sky, Surya Kant Jadhav, whoever. And then, you need not to think much more on that, because, I will be talking about, because the other 2 lectures which you have listened just now, mostly on basic sciences and basic research, particularly. And being a natural resource manager, I'll be trying to converge those particular science to practices as an extension soil scientist. So that would be my aim. And we worked with, and it was a great pleasure always when your group leader, your captain just spoke before you. And our group leader, when Professor H.B. Singh was there in Banaras University, it was always, it was a mesmerizing moment for all of us to work with him and to listen to him and speak after him. So this is the another addition which was missing, I think, in the series that happened today. So by this time, I think everybody will be in a consensus that, well, microorganisms performs diverse role, right? And these diverse roles on this particular planet, and most of which they are very important to make this particular art, which is, having many hassles are coming up, and to make this art a habitable, make this ecosystem a sustainable ecosystem. And once you are trying to do it, definitely you will be exploiting different properties of those microorganisms. As I was mentioning, that I am going to start on those consolidation, because when you have advocator of your technology, when you have advocator of trichoderma, definitely for one reason of your biotic stress, because Professor Sharma, Professor H.B. Singh, they always focused on particularly on biotic stress moderation. So definitely, but I am going to talk about something related to abiotic stress, and particularly with reference to nutrient, because this is one issue which need to be deliberated at a length. Otherwise, the way things are moving, definitely there are many things are awaited, right? So as I was mentioning, that many properties of these particular microorganisms, they are basically utilized as a low input biotechnology to solve various problems, whether it is related to environment, whether it is related to nutrition, whether it is related to food security, whether it is related to sustenance. And these wider aspects, when I am talking about all these wider aspects, definitely unravel the role of microbes and towards achieving a sustainable world. So next slide, please. And this is one of my favorite slide, the left one, the India, a major agricultural producer. And I said that, yes, Indian agriculture has made great strides. We have listened to those beautiful technologies, beautiful interventions of science, but definitely whenever you move to the ground reality, what we have achieved, this is impeccable. So this is the slide which I presented, I still remember, on the 200th birth anniversary of Charles Darwin and 150th anniversary of that particular book, Origin of Species. So that time, why I am mentioning this, because when I was presenting that slide as a young assistant professor in BHU, and when I am presenting today, the picture is almost same, and it has been magnified, to quite some extent. That means we have achieved huge, whether it is food grain production, you take any aspects of agriculture, whether it is horticulture. In horticulture, we are again surpassing even better than we are performing than what we are achieving in food grains. So in every area, either we are number one or number two. So this has been possible due to a huge committed and concerted effort of our scientists. So whether it is in the eastern part of India, whether it is the western part, in everywhere, looking at the diversity, Indian agriculture has made it a great stride. Next slide, please. And when we analyze this data, definitely what we could able to find out that this much of your India's food grain supply, it is coming from this Indo-Gangetic plain, where I am particularly staying now, right? In this particular Indo-Gangetic, this is also in an Indo-Gangetic plain, and which is the heart belt of your green revolution, and that actually made all these all these developments, right? And that has contributed significantly towards this particular India's, whenever we look about those India's food security, when we talk about today's India's food security, food grain production around 330 plus million ton. So this particular patch is almost 45 million hectare of your total cultivated land that has contributed like anything. And if you look at this, and if you look at those ecosystem and those cropping system which particularly exist in this particular region, mostly we'll be getting rice wheat ecosystem is always a dominance. And if we look at the total contribution, if you look at wheat, it is more than 70% of the production is coming from this particular belt, and rice is around 45%. Next slide, please. So these Indo-Gangetic plains, because I said Indo-Gangetic plain is a champion in contributing this food basket. But whenever we look at those different issues, emerging issues which are coming up, whether it is due to those yield fatigue, whether it is due to those deterioration or impairment of your natural resources, definitely we come up with this particular issue that you have an overproduction, stagnation, you have a decline in yields. The yield which was attainable maybe 10 years back, it's not possible. And even if it is possible, you need to incur a huge amount of resources. And the most important part with which I am actually aligned is natural resources, natural resource management, whether it is soil organic matter decline, and that has made a huge debate and huge concern. And you have your pest, your greenhouse gas emission, input use efficiency. This is another very important part, because ultimately these inputs are very, very energy sensitive. Whenever we talk about 1 kg of simple nitrogen, if we just convert it in terms of energy, this is something which is equivalent to almost 15 to 16 megajoule per kg. Means if you wanted to produce this particular 1 kg nitrogen, that is going to have an expenditure of this much amount of energy. So whenever and if your use efficiency, you don't have an efficient and intelligent efficiency is not there, and if you don't have a smart efficiency of your input, definitely you are going to lose a lot, particularly with reference to your natural resources as well as the borrowed resources as well. And profitability diversification is always an issue. And when our Prime Minister talks about the doubling of farmers' income, definitely, particularly this part of India is very, very important in making this particular issue, happening. Next slide, please. The before one. The before, yeah. So when I'm talking about this particular, area in the Gangetic plains, you have an agricultural intensification, and that has a long-lasting negative legacies, largely because of those particular agrochemicals. Although the amount of fertilizer which we use, that is not something even we are using from our less from when we compare with our SERB correspondent. If we even compare with Bangladesh, Pakistan, we are using less. But still, even whatever we use, there are some discrepancies. So negative legacies actually happen because of these particular agrochemicals and simplification of your cropping system, because you have more dependence, more reliance on these rice wheat cropping system, and which is happening from many decades, which is known as your monocropping. Next slide. And on this particular aspect, in BHU, we made a few studies, whether it is related to those physical deterioration, whether it is related to your chemical deterioration, whether it is related to your, microbiological or those microbial population deterioration that has happened in this particular patch. So there are, severe consequences and severe alarm we have noticed that particularly these particular patches is experiencing, whether it is with reference to your soil organic matter reserve, whether it is related to the nature of water movement through your soil profile, because that is very important. If you cannot ensure that particular movement and aeration is happening in your ecosystem or in your biome, definitely you cannot make it happen that it is going to do at its best. So we worked under these particular different segments, whether, as I said, that whether physical, chemical, and those microbiological aspects. And when we we deliberated on that, we have seen that there are huge impairment as well as deterioration has taken place, whether it is related to your conductivity of your soil. Because when I was talking about those infiltration and percolation, we have seen, there are many cases that has happened, that there are in many cases you have a water stagnation and you have an impervious layer which has been created, and that impervious layer actually resists your ultimate below ground bios, your above ground biosystem, or this below ground related to your root architecture is going to be impaired, root architecture is going to be affected, and ultimately your use efficiency is going to be impacted. So in a sense, what I am trying to emphasize here is you have a severe deterioration of natural resources quality that has taken place in this particular Indo-Gangetic plain, and that actually resulted, whether those particular yield stabilization or that yield plateau which we are experiencing, and at the same time the efficiency of our those added intervention is also very less. Next slide. So from, if we just look at this particular slide, you will be able to see that particularly from earthworm to nematode, from mites to insect larvae and fungi to bacteria, there are more than 7,000 types of species are existing in our soil. But if we keep on intensifying it with reference to those particular chemical inputs, whether it is fertilizer, whether it is related to agrochemicals, definitely at the end of the day, definitely we are reducing the biodiversity. Reducing the biodiversity, the soil biodiversity is going to be impacted. And as a matter of fact, whenever you don't have a good biodiversity, because we also worked out in that particular deterioration, in the previous slide where I have shown that it is not only physical, chemical, you have your microbiological deterioration that has also taken place, whether it is the population of actinomycetes, whether in some cases the population of bacteria that has been declined tremendously, even fungi. So and ultimately that particular reduction or that particular reduced biodiversity is not helping in this particular transformation to take place. Because ultimately, whenever you talk about any efficiency of your nutrient, you have to talk about the source as well as the sink. So definitely, whenever you talk about the source and the sink, you have to talk about the dynamism of there. The dynamism may be in the total source, maybe in the available source and your soil solution form. And these all mediator of these processes are all your microbes, this particular force, and who actually works day and night 24/7. So next slide, please. Next slide. Yeah. So ultimately, when we talk about this particular soil, because this particular soil is a non-renewable source, and this is very, and when you look about the basis, whenever we look about our existence, whether it is for feed, for medicines, ecosystem services, well, for everything. So these particular resources, and that has been, intricately related with your all attaining your sustainable development goals. So you take any goals of your sustainable development goals 1 to 17, either they are directly or indirectly related to this non-renewable resource. So there are some impairment which has been taking place, and that is continuously, if it works like this, definitely there will be a disaster. Next slide, please. So the challenge is, we have already, we are appraised with this, that the population, the things, it is moving definitely for, we achieved with the, the good amount of production. But now we are talking about your quality, the health and nutrition is getting more importance. But these agricultural, whatever production we have achieved, so this has at the cost of impairing your soils, impairing about this particular natural resource base. And this is under tremendous pressure of intensification. And as I was mentioning about those competing uses of those resources, because ultimately whenever rendering those ecosystem services, we are not able to, cope up with the challenge. Next slide, please. So there are 10 traits. Basically, whenever we look at as a natural resource manager, whether we work in these Indo-Gangetic plains or the rest part of the world, definitely from 1 to 10 of this particular problem, there will be many. Or in many cases, it may so happen that, the 10 problem will be there, they coexist. So for our case, we have a organic carbon change. We have a depletion of your soil organic carbon. And that actually, because of the subtropical climate, because of this, the rate of decomposition, the way it is happening, and rate of disturbance of your soil. So that actually, whether it is through your, mechanization of your agriculture, so that actually impacted heavily on this particular organic carbon. And as a matter of fact, because this organic carbon is basically your driver, so these actually drives most of your nutrient cycling processes. And if it is, driving those nutrient cycling processes, definitely the next thing which is going to impact is your nutrient imbalance. And that has actually in many part also of these Indo-Gangetic plains, you have the issues of salinization and solidification. So in many cases, this salinization and solidification, it's not something anthropogenic in nature. These are all geogenic in nature. But still, there are some cases, we have some reports, we have some deliberated, attempt of this human force to make those particular, soil natural base, to make them, convert them to saline or sodic. So that is also happening because of intervention or addition of those input material. So these are the issues, particularly there are, as I was mentioning, those 10 threats, but particularly for Indo-Gangetic plain, we have these particular issues which are giving us, a huge amount of agony. Next slide, please. So 33%, as a matter of fact, this particular, whether, we look at globally, whether we look at regionally or locally, your soils are being degraded. And when you have soil degradation, when you have soil impairment, when you have soil deterioration, definitely we need to make some sort of intervention, because many a time you cannot talk about some rocket science here, but definitely some intervention, some of your intervention, whether it is bioaugmentation, whether it is some sort of intervention, introducing some microbial or microbes or microbial consortia to a particular ecosystem, can help you a lot. Next slide, please. So this is another very important, very important and very favorite slide of me, that I always like to talk about this, that soils are like books, it is always judged by their cover. The chapters which are thereafter, nobody looks into it. And ultimately, whenever you just put on the cover, you see the things are happening, whether it is with reference to the color, with reference to the vegetation, the above ground vegetation. So that actually, that is very important. And those layers like chapters are often not read all the way through. They're an immense resource, yet commonly they're undervalued. That means what I'm trying to, pinpoint here is the below ground biodiversity, which is going to make all those, impact, all those ramifications, as well as manifestation at the cover pages, that is still undervalued or underestimated. And when I was talking about those 7,000 species and those the helpful organisms, or those particular isolating those particular microbes, which is for that particular agroecosystem, which is specific to that particular ecosystem, because when we talk about microbiome of a specific crops, you can, in a generalized way, microbiome of a particular ecosystem, or those rice ecosystem, wheat ecosystem, that can also be talked in detail. Next. So this is next. This is an ideal system. This is an ideal system that what it should be, means the way we are introducing something into this particular ecosystem, or this natural ecosystem, or this particular soil, and whatever we are taking out from this system. So you have to be a parity. You have to be a, if there is any discrepancy between this input and output, and then things goes beyond your control, and then you have all sorts of hassles will be coming out. So whenever we talk about sustainability, whenever we talk about sustenance, definitely this particular dynamism, not only with reference to the energy inputs, what energy we are including into this particular ecosystem, and what energy we are taking out of this particular ecosystem. That has to be calculated with more authenticity, and that will give more comprehensiveness. And this is an ideal system, as I said. And if it should match, means the input should match with output. If it doesn't, next slide please. If it doesn't, then you will be having I was talking about those planned nutrient balance system next. So if it doesn't happen, then you will be having that sort of mining to take place. So that is happening in this particular Indo-Gangetic plain. So you have your removal of your crop residues, the reliance and dependence on your organics is getting, we are just far away from those organics, and you have a skewed NPK ratio, and it is mostly always a biased fertilization is always being done. So it is always nitrogen driven economy. It's a nitrogen driven application. Mostly it is a nitrogen skewed one. So, and this is a conceptual framework, as I was mentioning, because if you have a discrepancy that what you are adding and what you are taking out, so definitely you will be creating a negative budget. And if you going to do that for a number of years, number of consistent years, definitely that is going to be widened. And if it is going to be widened, definitely plant will be in at a helm of affairs of taking those particular nutrients from that particular soil or from that particular ecosystem at its the maximum resources or with the maximum energy. So next slide please. So, and this particular area is also when we started, because when I was showing that particular patch, I was talking about those deterioration. We also worked out those chemical properties of soil to find out that, well, whether there are any discrepancy with reference to the desirable or the acceptable or the content or the available content, which you can make a synergy with your plant requirement. To our surprise, we found that even till 2010, in many places, there were no report, particularly I'm talking about these Indo-Gangetic plains and the adjoining areas of this particular Varanasi district. When we were working with those adjoining Varanasi districts and Varanasi as well, we found that there are issues like sulfur, there are issues like boron and zinc, which the people before 2010, there were no report that yes, there are deficiencies are coming up. So there are multiple multi-nutrient deficiencies are coming out. And these multi-nutrient deficiencies are coming up because of that sort of sophistication, that sort of biasness towards a particular input like nitrogen. All of our advocacies are always related to your macronutrient. We don't advocate for your micro and secondary nutrients. So where from it is going to come? So if you are not applying, if you are not substituting, if you are not incorporating, definitely there will be a difference. Next slide, please. So as a matter of fact, this particular system is being overloaded. With reference to intensification, with reference to the huge load on a particular group of people. It's just like working in a government office. Once it is being somebody gets retirement, there is no appointment like that. So that particular population of those drivers, that is going less like that. So and those particular population who are mediating this, definitely that has to be restored. So what is the message? That we need to look at the below ground biodiversity with more comprehensive details, and that the ground truth details need to be we need to have those ground truth details, that what is happening in and around in those particular ecosystem, in those particular, whether it is a rice wheat, whether it is a other vegetable based trophic system or like that. So we need to look at those below ground biodiversity. We know that 1 gram of soil can contain the population of the whole universe or more than that. So definitely it's a huge powerful one to mediate all those processes. Next slide, please. Next slide. So from the previous one, what we can derive out of it. Yes, definitely we need to make some rhizosphere manipulation, and this rhizosphere manipulation becoming the missing link between what we wanted to achieve and what we presently achieving. And this, right, when we, if we wanted to achieve that, particularly with reference to whether it is with reference to input use efficiency, whether it is with reference to sustenance, attaining the maximum potential in, we need to have a rhizosphere, an active rhizosphere, a vibrant rhizosphere, and with a higher efficiency. And if it is that, definitely we have to make a synergy between those resources, as well as Professor H.B. Singh sir was mentioning, that it is, in the present context, it's not possible to go for 100% organic or 100% bio-based economy cannot be there. But we have to do those sort of compromises. We have to come up those particular recipes where we, even without compromising, we can achieve that particular potential yield, because we have to feed this huge population. Next slide, please. And that particular from these particular deliberation, one thing is very pertinent, that yes, this particular this conventional techniques, they have degraded our soil quality, particularly these endogangetic plains. And beneficial microorganism can partly, as I am giving a specific emphasis on this, that partly substitute this high quantity of or the costly chemicals required. Because when you talk about fertilizer, the amount of subsidy which government of India is giving, if you look at even 2022, 2023, it's more than 1 lakh crore subsidies being given, right? So definitely it's a huge, as I was mentioning earlier, that it's a huge energy sensitive. As I at the same time I said, a huge amount of subsidies being given. That means always government is also working out to find out some compromises so that the subsidy can be reduced and we can introduce these particular beneficial microorganisms without any compromise in terms of your production. So bioinoculants have been identified to release these bioactive compounds helpful for plant growth promotion and stress mitigation. Because my objectivity will be, when I will be looking out those particular results which we have obtained under this particular ecosystem, every time I will be giving emphasis on your nutrient. That means those abiotic stress moderation or mitigation. Next slide, please. Next slide. So the question which we wanted to raise is definitely, can we make some simple intervention? Because many a time, as an extension worker, as an extension soil scientist, when we visited field, or when we used to visit, because we made it a practice visiting field. So every time you cannot make your intervention straight away. So whatever is existing within that particular ecosystem, with that particular system, can you make some simple intervention that can give you, or that can assure you that you have some system without much dependence on non-renewable energy to save a reasonable amount of subsidy, as I was mentioning, and to improve your nutrient use efficiency. You can improve your system productivity and impairment of your environmental resources, particularly when this particular endogangetic plain people talk about the greenhouse gas emission because of those burning of those residues, that is also coming up. So can we have any a simple intervention that can work and that can work for our farmer? Okay, next slide, please. The legacies at work, that this plant soil microbiome interaction, definitely it is underpinning your agricultural sustainability. That I'm going to show you in next few slides, that these legacies definitely will work. And this is this was working earlier also, but in between we missed those linking, we missed those we made some sort of intervention which didn't like these legacies of chemical dependence. Because here I always mention one thing, that there should be a marriage between chemistry and biology. Chemistry cannot work independently, or biology cannot work independently. You have to make a marriage, otherwise the system is going to be divorced. So the legacies has to work. Next slide, please. Next slide. So there is now increasing attention of creating a positive and above and below ground legacies through selecting crop species genotypes or optimizing a temporal and spatial crop combination. We are not going to talk on that, but improving your nutrient inputs or efficiencies, as well as applying soil or microbiome inoculations. Professor Sharma was mentioning about those, inoculant or mixture of or inoculant consortia that we have worked. What those Professor Sharma and his lab and Professor H. Vishing, when he was here in BHU, what they were developing, we were the force who actually, worked, in implementing, or better if I tell like this, that we try to convert those hardcore science to practice. Yes, next slide, please. So this is a conceptual model of biopriming, which we actually followed, particularly in our case. We introduced that particular bioprimer, those inoculant, either it is a single inoculant or maybe a consortia. And then we actually, with those existing practices, we just make a simple intervention with reference to priming. And this is, as I was mentioning, these bioagents, maybe something who is, capable to facilitate or mediate the process of nitrogen cycling, maybe someone who is, capable to, make a mediation in phosphorus cycling, maybe potassium cycling. Recently, we also worked with sulfur cycling also. So ultimately, with that objective of your mineral nutrition and with a more use efficiency, bioactive compounds definitely there will be there, and your stress tolerance and crop yield. Whenever I will be, talking about those particular farmer field experience, there we have experienced that this particular stress tolerance of, adapting at a variable vagaries of weather, because, we are at the exposure of that particular situation. Farmers, they don't know that whether the winter is going to be a prolonged winter, or whether there will be a, prolonged rainfall, or there will be a, suddenly you have a stop or withdrawal of monsoon. So to address those issues, whether this particular, technique is capable to address like that. Okay, next. So initially, we started working it in our labs, in our university farms and the adjoining areas. So this is basically what I was mentioning, the biopriming with those bioprimer. This is the simple mode of mixing those bioprimer using your normal seeds, and it's initially these were those the initial study where we could see that you have a, the below ground biomass. If you look at those particular root growth, root architecture, and the root volume and the root mass, everywhere, you have an extension, an expansion, and that can explore a larger area for exploring or those particular nutrients to take from a, larger distance. So definitely things have been improved. Well, next slide. So adaptive, because, whenever you talk about this sort of intervention, that has to adapt with that particular ecosystem. That means you have adaptive soil management has a broader benefit, definitely, when you interlink with other natural resources. And definitely it is going to combat your regional challenges of your sustainable agriculture, particularly attaining whether it is sustenance of yield, whether it is, sustenance in your environment also. Next. So this is, as I was mentioning, maybe from single specific soil microbes to a more holistic microbiome approach. Things people are working on it. Professor Singh mentioned that, there are one company which came up with that sort of consortia. So, and definitely under this particular, direction, people are concentrating. Even Professor Birinchi Sharma sir is also working on that direction. And we have, in our experiences, taking out those particular isolates, and then we also worked out those particular synergism. We also worked out about those compatibility in our lab, and based on those compatibility, then we select those consortia for application in the field. Next slide. So paving, as I was mentioning, it's a paving the way from the lab to the field and developing those microbial consortia, and then working out with those microbial consortia, and then finding out their their compatibility, because that is very important, because that is one issue. Even if you don't have whatever the inoculants are available in the market, even you can, if you know that this particular species is, capable to have a broader interaction and broader synergistic interaction with one species, definitely you can try. And even in our lab also, we attempted in that particular fashion also. So it's a journey from what Professor Sharma and his co-workers and his team is working, and we are just taking that particular either a single microbial culture or maybe a consortia, and for looking out the effect on your abiotic components. Yes, next. And this is our experiences, as I was mentioning, in adjoining places on farm biopriming. And as I said, that is a simple, robust, widely applicable technology. And these are those adjoining districts where those, inoculation or those particular, priming intervention has worked like anything, with reference to its yield, with reference to its nutrient use efficiency, the growth attributes. But, those particular intervention was always made, as I was mentioning, that you can partly address the situation. That means if you have a recommended dose of fertilizer, whether it is by the government of Uttar Pradesh or by Indian Council of Agricultural Research, if they have something like 120-60-60, so at what extent you can, just cartle down, maybe from 120, if we can make it, 100. So these 20 kg, substitution by this particular priming intervention, that actually we are looking for. And yes, in the next slide, we'll be looking at some other crops. We also looked at some flowering crops also in our institute. Next, we also with some high value crops like red cabbage, because this is something, because, mostly in our study, we focus mostly on those crops who have a high fertilizer requirement. Means, looking at that particular point, we actually emphasize those study that whether, when I was mentioning here, you can see 75% RDA plus trichoderma, and then fluorescence, and so that particular gave almost equivalent, to the non-prime plants. So that means, you can straight away reduce 25% of your fertilization, right? And we have sent this particular report to the government of Uttar Pradesh for their adoption in their, Kharif meeting. Okay, next slide, please. So basically, I'm not going to, look at all those studies which our students made in the campus or the off-campus, but at the end of the day, we are basically convinced that what is happening, definitely, either very recently in 2023, this month only, we also published one paper where we proved that this trichoderma is capable for your faster reserve mobilization of your, from in the seed cotyledon, right? Faster reserve mobilization of in the seed cotyledon, and that we have proved by, analyzing many enzymes. So this is, and also Professor Dwivedi and Professor Sharma, they have also worked on those upscaling of those particular, primers, and those which is giving the more efficiency of those, nitrogen moderating enzymes that are actually implicated, and that has actually, that actually we have observed in our study on your above ground biopart, whether it is in your vegetative part or whether it is in the reproductive part. Means whatever nitrogen is coming up or whatever, other nutrients which has been transferred to your reproductive part, that has been proved. Okay, so that means we are convinced. Next slide, please. So the next question is always that whether, how far it is going to work under field condition, because, Professor H.B. Singh has trained us in that way that yes, whatever you are doing, whatever you people are doing, you should be, feel convinced until you reach to that particular last man in the row. And if you are visiting them, if things working, whatever you are advocating, if it works for them, then you should feel happy that yes, your technology, your intervention works, right? So now we will look at that what is happening in farmers field. Okay, next slide. So here what we done, because Mera Gao, Mera Gaurav, there's one, I think many of you might be knowing that Prime Minister, this is a dream project was there. So in that, I took advantage of that, and there I sensitized, means personally I visited them, and we as many of my students also involved in that. So we sensitize one farmer club, and they have a huge farmer club in that particular part of Bengal, and this is also comes under the Indo-Gangetic plains. So and there we just introduced with this delivery system. And in that particular farmer club, they have their FPOs, they have their SHGs and everything they have. And we also make it a point that, well, we will train them, as well as we will try to make a gender neutral world also, so as to make that the woman farmer also empowered, because many a time whenever the seed treating business used to be done at the farm level, definitely in most of the cases the farm women were involved. So we take care that particular part. So we sensitize one farmer club. Okay, next slide. Next slide. So this is that Krishak Ratna Farmers Club, and we started this venture during pandemic, when the entire world was challenged. So that particular time we actually took this initiative, because we were there in the our closed room, and we tried to disseminate the our strategies, our intervention. So I physically went to those particular area, trained them, even involve the local administration, that whatever we are doing and for what reason we are doing. And at the same time, we also just like what I am doing it today, so online intervention was also there for making them educated that this is a simple intervention, and we are going to send those culture for you, and you have to introduce that particular culture to your existing system, and to go ahead with your particular cropping intervention. Next slide. And this is that particular area. Next slide. And you can see those intervention. I was because whenever we are introducing this particular species, definitely we have an idea of augmenting that particular species in that particular area, because this bioaugmentation of your diverse soil microbiome, definitely it will promote your sustainable agriculture and can effectively contribute to meet your triple requirement. And for rice and wheat, we persisted with a single culture, means your trichoderma. But whenever we started very recently, but that data is not still with me, we started with maize also, where we started with the broth culture of co-inoculants. We send those culture, and the last time when I visited them, I also carry those culture with me, and I have given them. So they work with that particular crop, maize also. Okay, next. So co-inoculation, I'm talking about that co-inoculation. Okay, next. So that was that convergence I was talking about. So the only intervention was made, during the step number 5. You see, the rest is everything was same, what they were doing. The only thing which we wanted to make that during your seed treatment, right? Seed treatment. And in that seed treatment, we just introduced those particular inoculant to them. We have made them available, that particular inoculant, and they have just mixed those particular inoculant for a particular specific hours, and then they do the job. Yes, next. And I was talking about the gender neutrality, because there were mostly the SHGs were run by women farmers. So there, because I was talking about addressing these climatic irregularities, right? So that part of India, even this part of India also, they are not, many a time it may so happen, and it is happening, that during Kharif season, they don't know that suddenly during the seedbed preparation, you have a sudden flood is coming, or there may be a withdrawal. So that particular seed tray technique is very popular now at that particular, that becomes a seedling factory. And this seedling factory, these all seedlings are being treated with those inoculant, and now they are procuring it from the local university, local state agriculture university. And they are just, treat those, you can see those seed beds, they are having those seed beds, because you need not to, make any other intervention related to preparation of seed beds. So you can come to your factory for seedling, and you can go ahead with your, field with those seedling, which is inoculated with your, for your transplanting, or which is inoculated with those inoculant, and which is ready for transplanting. Yes, next. And the difference is crystal clear. This is some photographs which has been sent by some of the farmer. So they have they have been also are convinced, or they have been also able to identify the difference of those underground biomass, because this is a key player. This underground biomass, whether you are going to recruit your microbes over there, or whether your root is going to proliferate or will attract some of your other microorganisms because of their secretion or secretion of some of your root exudates. So everything is going to be centered around this below ground biomass. So this below ground biomass architecture is also being changed. This is one can change with his naked eye, that yes, things are very different, and this difference is quite considerable. Yes, next. And now I am going to talk about that. Well, because we took help of the local state agriculture university, where our samples from farmers field that has been they have observed that, and whatever data they have sent, I'm just so you can see there are changes. There are changes of for those particular whether it is through your bioaugmentation or whether it is through your biopriming or that particular intervention, you have a change, because I said that is a rice wheat ecosystem. So we collected samples after the harvest of rice as well as after harvest of wheat, and we have seen the population of this bacteria and fungi. They could only see this particular population, and you can see we are drafting a paper on this, so that's why I did put all the details over here. So the only thing I'm going to tell over here is there are changes, that there are changes with reference to the diversity. There are changes and with reference to the diversity, whether it is in the aerobic system or whether it is anaerobic system. Whenever you are going to introduce your rice, definitely you are inculcating that sort of anaerobism in your anaerobiosis in your soil, or anaerobiosis in that particular ecosystem. And whenever you are going to introduce your wheat, definitely you have your aerobic populations and all that, and they differ. Okay, next. And if we look at, because this is my area where I actually give more emphasis, that is your physical and chemical properties of soil that has changed. That has changed. As I was mentioning that yes, we started this program in 2016 initially, and then it has been magnified during the corona period. So we have changes. You can see the changes with reference to bulk density, whether with reference to this mean weight diameter, because these soil aggregates, whether it is making whenever the proliferation of your roots or the microbes proliferation, microbe population, to enthuse those population, you need to have those the diameter of that particular soil particle should be such that so that it can help for either your proliferation of roots or or taking out those particular cycling process to happen. So in these cases, although the differences are not that very, because we have done those statistical analysis also, the differences are not that statistically significant, but there are there are positive changes with reference to physical properties, because physical property is very difficult to change in 1 year, 2 year, or 3 year. Definitely it needs a decade or maybe 5 years, 6 years, 7 years to make those changes. But there are, there are some changes, positive changes are happening. Next, we'll talk about the chemical properties. Chemical properties, that is with reference to nitrogen. If you look at your phosphorus, so that has changed. Once you make that sort of intervention. And definitely the availability, whether it is at your soil solution form, because that is the most important form which we look for, not only not the available form, because ultimately that is the form which plant takes with ease, without any difficulty, without any hassle. So that with reference to those nitrogen, phosphorus, and potash, there are the changes that have taken place, particularly with reference to phosphorus. There are significant changes, I can tell, that has taken place. With reference to nitrogen also, there are changes, positive changes. Even potassium also has made changes. And now I will show you which is the data which was taken, because these data were mostly taken up by those, as I was mentioning, State Agriculture University students or scientists. But now this data which I am going to show you, that has been taken up by the farmers, means who are implementing that particular intervention. Yes, next. So if you look at this data, next slide, please. If you look at the grain yield, because ultimately every time our objectivity is whether we are capable to attain those potential yield or not. Okay. So you can see here that, in 2018, it happened that there are withdrawal of monsoon that take place at that particular area. Means there was a continuous dry spell for almost 45 days. 45 days, just imagine, 45 days there was no rain. So you have to think for those exploitation of your underground resources. So that happened, but many farmers, they cannot afford that. So that in that year, if you look at without priming and with priming result, you will be able to see the difference. That without priming, that particular plant was capable to adjust or accommodate under that particular harsh condition. And it also happened in 2019, where in your wheat crop, where it is a necessity, or it is
Dr. Rakshit,
yes, I'm
we are
just
crossing the time limit, please.
I'm just I think 2, 3, 4 slides are there. I'll be finishing first. Okay, so in case of wheat, whatever we have seen that, okay, as I was mentioning, that you cannot sustain with a shorter winter. You need a longer or prolonged winter. You see 2.85 and 3.65. So there is and this is the initial trends was there, and this has been reported by those farmers. Okay, next slide. So anything which is small, but small can be always beautiful, because we only intervene in the process of seed treatment, but we are getting some positive result. Okay, next. So what is evident? The evident part of this story is that development practitioners are also increasingly recognize the need for their collective action. We involve those administration, we involve those SHGs for adoption of many things what we wanted to convert. Promoting sustainable natural resource management, you need to require an understanding of this interaction of this local external institution, and you have to work on this building on this local strength. And microbiome based innovations can contribute to policies that interface with this sustainable development goal, and cooperation in microbiome science is crucial for success. And there is a need to embrace agricultural practices that do not depend on greater use of fertilizer, because even when the present government, the paramparagat krishi, the organic agriculture, every time we have very recently in the BHU, we met for a particular program which is known as Shufalam. People from across India came. We try to formulate or try to just inventorize those technologies which can can substitute some part of your this inorganics, right? And water to meet the growing demand of global food requirements. An effective microbiome can offer benefits to host, including plant growth, promotion, nutrient use efficiency. Next. So the last part is this continuation of this rice wheat ecosystem. It is going to particularly this part of India we have, and that's why I was showing those some high value crops, and we have been we are also working on that to convince farmer, and there are many farmers. They come up with some simple innovations of replacing this particular ecosystem, which was responsible for making all this deterioration. Biopriming or this bioaugmentation has a huge potential in restoring this present setup of this fragile, and successful program management is here dependent on awareness raised regarding the importance of application of these eco-friendly practices. Okay, so I'm going to I'm very next slide. I'm thankful to those collaborators as well as the funding agency who has always helped us in achieving these small experiments, whether it is in BHU or whether it is in the off-campus in farmers field. And so definitely they have made it possible for this particular task. Next slide. And everything was possible because of this extended team who actually work in lab day and night, and they came up with those particular studies and those results, and which actually I presented and summarized. And I didn't mention that particular a specific paper in a sense. I was just trying to put it in a generalized way. So thank you very much once again, Professor Chaudhary, for giving me this opportunity. And I'm very thankful to the audience as well for this hearing this particular deliberation. Thank you very much.
Thank you, Dr. Rakshit. Any questions from a student or faculty colleagues? So if we do not have, we express profound gratitude to Dr. Rakshit. Thank you, sir, for nice talk, and please visit Noida next time.
Thank you very much. Thank you very much.
Thank you, sir.
I extend invitation for your group as well to visit BHU Varanasi, because our representative Professor Sharma is there, and I think he is taking care of that part as well.
I call those brain over here who are working in biological agents in agriculture. So it was a theme in my mind. So how do we can deploy microbiome in mitigation of the climate change to mitigate the perils of the climate change? So it was very deliberate at my end. Thank you, sir. So nice of you. So we will.
Thank you very much. Thank you very much.
Thank you very much. Now we are proceeding ahead with the last colleague, Dr. Anukul. Ayushi, please.
Now we would like to invite Dr. Anukul Vaishnav for his talk on plant communication with soil microbes for asking help under stress condition. He has devoted his whole research work in the area of microbial intervention in agriculture for enhancing soil health and crop productivity under stress conditions. He has completed his doctoral research in the area of plant microbe interaction. Recently, he has completed 1-year postdoc in University of Zurich, Switzerland, as Swiss Excellence Postdoctoral Fellow supported by Federal Commission for Scholarship, Switzerland. Currently, he is working with GLA University, Mathura as assistant professor. In his research work, he investigated how soil microbes improve plants' immunity against abiotic and biotic stresses. His results demonstrate that beneficial microbes and their metabolites enhance plant survival by regulating defense gene expression, iron homeostasis, phytohormone signaling, and nutrient uptake. His recent results revealed that mycorrhizal fungi act as a mediator of disease resistance in maize plants. Dr. Vaishnav has published more than 50 research articles, including book chapters, and edited 8 books in leading scientific journals and publishers. His research group recently granted an Indian patent on a novel biocontrol agent for controlling fungal phytopathogens. Dr. Vaishnav has been awarded different prestigious fellowships like DBT, ICAR, SERB, and Swiss Excellence Fellowship, and several research awards from reputed societies. We welcome you, sir.
Thank you for your nice addressing. I'm I'm pleased to be here because this is my alma mater, and it's always about the pride if you present your work in front of your supervisor and your mentor. So I'm thankful to Chaudhary sir for inviting me here. And I know that you all are in hurry, you want to go back to home, someone are going to back, and many of you are waiting for the tea. So I would compile fast. Here I mention my topic, that plant communication with soil microbes for asking help under stress condition. I would try to make it interesting. You always learn about the plant communication, like how plant communicate to other plant, right? Hopefully you learn in your BSc and MSc, but here I will talk about that how plant talk to soil microbes. How plant talk to soil microbes. If you imagine, you will find it interesting that how plant will talk to soil microbe, because they can't say hello, hi, how are you, but still they talk to soil microbes. That will we will see in the next slide how plant will talk to soil microbes for asking the help under stress condition. This is the slide that I always use in my presentation because this is the need for research on the agriculture in sustainable agriculture. Here you can see that out of the total land, we have only 29% land for the agriculture. And in the last column, you can see that out of 29%, only 23% is used for the crop cultivation. Only 23% out of 29%. So what we have the challenge, we have limited land and we have to improve our productivity on this limited land only. We have to feed our growing population by using only this limited land. That's why we are working on the agriculture field. That's why we are working on the sustainable agriculture. These are the rationale that everyone discussed from the morning, Sarma sir and the Rakshit sir and HB Singh sir. So I will skip this. I will directly come to the interesting part that you want to listen, that how plant communicate with the soil microbes. So I will skip some slides and directly come on this part. So these are the basic things that you know, that soil microorganisms are responsible for the nutrient transformation. And these are the 10 to power 9 and 10 to power 6 bacteria and fungal taxa are present in the soil. The interesting thing is the plant release 20% photosynthesis in the soil. You know that plant use, when plant make any carbohydrate, when plant use photosynthesis, plant expend 8 ATPs, right? I'm right, 8 ATPs for synthesizing 6 carbon carbohydrate. But it released 20% photosynthesis into the soil. After doing all type of mehnat, it released 20% of the photosynthesis into the soil. If I ask to someone in front of mine that, can you donate your 20% salary to your neighbors? You will say, what foolish you are talking about. But plants always do such type of things. Plant donate 20% of their salary, that is photosynthesis, into the soil. And that is best thing. There is a statement in Hindi, दान दिए धन ना घटे, कह गए दास कबीर. अगर दान देने से धन नहीं घटता है तो आपको दान देना चाहिए. तो plant do such type of things. Plant release 20% of photosynthesis into the soil for attracting microbes that can help to plant for survive under stress condition. That is the communication way. How plant communicate to microbes. That is the thing. Plant release photosynthesis, plant release carbon compounds, plant release amino acids that we call root exudates. So plant release such type of compounds that attract beneficial microbes towards the rhizosphere that help plants to survive under stress condition. And that is called cry for help strategy, that defined by the Sarma sir in the morning section. So here I mentioned some compounds that is responsible for attracting beneficial microbes. That are compounds are benzoxenoids, that is well reported in the case of maize, methyl salicylate, methyl jesmonate. These are the compounds that is reported to attract beneficial microbes. So this is the way that how plant communicate to microbes. Plant have to release some interesting type of compounds that can attract beneficial microbes towards the rhizosphere, or we can say towards the plant environment. And these microbes help for plant survival. This is the mechanism, molecular mechanism. We can say that how bacteria, how microbe attract towards the root. So here you can see once if you can see the pointer, here root is exudates some compounds that is attracted by the microbes. How can this one? Okay. So here you can see that the plant release exudates in the pink color and it will attract the beneficial microbes. Okay. And this root exudate will be received by the receptor that is present on the bacterial cell wall here. Okay. And once these microbes attach to the root surface, they will start signaling inside the cell. Here you can see the red color. And they will start to form biofilm on the root surface, like here you can see. And this biofilm, here they present that this biofilm can show as a role as an antibiosis, like they can inhibit the growth of fungal pathogen. These are the pathogen, and they can work as a resource competition. Like they will never allow to grow other pathogenic microbes as the form of the resource competition. So this is the molecular mechanism when microbe attaches to the root surface, when microbe attract towards the root, plant root, this is the molecular mechanism. And we try to find out this molecular mechanism in our research work. So this is my first experiment where I try to find that how root exudate enhance biofilm formation. So here you can see that this one is the control condition and this one is the treated condition. Treated means root exudate secreted by in the presence of salt stress. I mentioned in the legend, soybean root exudate under salt stress condition. So this is the treated one. So you can see that the more number of biofilm, or you can see the more concentration of biofilm produced in the presence of treated root exudate. This is the interesting study. You can see here that the root exudate enhance biofilm formation of the bacteria that we see by a simple assay, by microtiter plate assay. And this is again the interesting thing. When we try to find out the exudate compound, what compound are responsible for attracting microbe, what compound are responsible for the enhancing biofilm formation. Here you can see that the green one are the newly expressed compound in soybean root exudate when we treat our soybean plant under salt stress condition. So you can say that these compound, the green one, aspartyl lysine, aspartyl isoleucine, these green one are responsible for enhancing biofilm formation and for attracting microbe, that is Pseudomonas simei au. This is the PGPR, plant growth promoting rhizobacteria, that was attracted by soybean root exudate and in the presence of salt stress condition. So here you can see that how soybean root exudate communicate with Pseudomonas simei for asking help. And in response to root exudate, Pseudomonas also secretes some beneficial volatiles. This is give and take mechanism. If plant releasing some exudate, then in response to exudate, the Pseudomonas simei also release some beneficial volatiles that help for plant promotion. You can see in next slide. And these volatiles you can see like hexanol, nitroglycol, they are well reported as a plant growth stimulants. And tridecaphenols and benzothiazole, these are reported as a biocontrol activity. These metabolites released by the bacteria, once they are attracted by the soybean root, then they will release such type of volatiles for improving plant health and inducing resistance. So here you can see that this Pseudomonas simei improve plant growth under salt stress condition. That is, I mentioned contact-less interaction. Like in our earlier days, sadhu आशीर्वाद देते थे, तपस्वी भव, दूर से ही आशीर्वाद देते थे. यही केस यहाँ पे हुआ है. You can see here that we inoculate our seed here and we inoculate our bacteria on the lid. There is no contact between the bacteria and seed, but still bacteria is able to promote plant growth under stress condition. That is because of the volatile exposure. That is because of the volatile released by the bacteria that is present on the lid. There is no physical contact. That's why I mentioned here contactless interaction. And these are some functional analysis, molecular mechanism, like when we expose our plant with the volatiles, they will induce some phytohormones, ethylene, salicylic acid, abscisic acid, that is somehow responsible for the stress tolerance. And here, if we talk about the drought stress, the volatile modulate transporter for water, like this is PIP and TIP, this is for the water transporter. So they modulate this transporter expression. So what plant can stand under drought stress condition if we expose our plant with the bacterial volatiles. And these are some antioxidant analysis that we perform under page. This is for polyphenol oxidase, superoxide dismutase, and peroxidase. Antioxidant enzyme, you all know about antioxidant enzyme. They will protect our plant, they will protect our cell from reactive oxygen species. So if we put our bacteria, if we put our microbe that release beneficial volatiles, that volatiles protect our plant with ROS, reactive oxygen species. And these some proteomic study. Here you can see that the bacterial inoculation increase thiamine production, catalase production, ATP synthase production. Elongation factor 1 alpha, that is for translation process. So these proteins increase in the presence of our bacterial inoculation when we inoculate our bacteria and when we inoculate our bacterial volatiles in our plants. And this one is for the biotic state. This was also interesting study. You can see here we use 2 compartments. Here we inoculate our microbe, trichoderma, and this one is the plant pot. And this trichoderma is connected with the air pipe. See how it is interesting. This microbe pot is connected with air pipe. There is no physical contact between microbe and plant, but still this microbe help for plant growth and for disease resistance. Now you can imagine that these microbe, this trichoderma release such type of volatiles that are in gaseous nature and that induce resistance in this okra plant against fungal pathogen. Here I mentioned that. So we perform first in vitro study. This is our trichoderma that inhibit the growth of Fusarium oxysporum. And here you see the one other pathogen is S. raultsi. Here you can see that our trichoderma degrade the hyphae of S. raultsi. And it also promote the growth of root in okra plant. But the interesting thing is it is not directly contact with the plant. Only volatiles is connecting. This is the communication way. Only volatiles is communicating with the plant and it improves the root growth. You can see here. And we identify different volatile compounds in trichoderma. This is the list. And this is my latest study that I performed in Switzerland. So here I try to find the role of mycorrhiza that is mostly reported for the nutrient uptake, right? You always read about the mycorrhiza is nutrient mobilizer, nutrient transformation. But here I try to find that how mycorrhiza help for disease resistance, how mycorrhiza can work as a transmitter between plant and soil feedback. So this work I performed in Switzerland and I found that the mycorrhiza hyphae that attract some specific type of microbes in the soil. Hyphae, not the root. Mycorrhiza hyphae attract some specific type of microbe that help for disease resistance. That you can see in next slide here. Like here you can see that this pink one, I analyze this on the basis of the family genus on the basis of genus. So you can see here that when we inoculate our mycorrhiza and when we infect our plant with pathogen, the change in the taxa belongs to Microciliaceae, Pyruliaceae, Xanthomonads. You can see here. So finally, I conclude that Sharma sir and Chaudhary sir also mentioned that with the help of microbe, we can chase the sustainable developmental goal. So here I mentioned that if we use microbes in our agriculture system, if we work on the microbial formulation, we can chase SDG 2 zero hunger, SDG 1 no poverty. So here I mentioned that for zero hunger, we can increase farm productivity if we use biofertilizer and biopesticide. We increase farm profitability. That can chase by SDG 1 no poverty. Because if we use biofertilizer and biopesticide, that is more cheaper than the chemical fertilizer, and the growth and the productivity will increase. So that will definitely increase the farm profitability also. And for SDG 3, 6, 13, and 15, if we use microbes that reduce the greenhouse gases, that reduce the climate impact, that reduce the climate changes, that is the main theme of this conference. And if we more produce bioformulation, then it definitely creating new industry for the production of microbial products and formulation. So it will also chase the SDG 8 goal. These are some books that we edited, and these all are about the beneficial microbes, that how they can help for plant growth promotion. This is all from my side. If you have any question, please ask.
Any questions from anyone? Students? So, yeah, I'm coming to you, Dr. Naveen. I congratulate Anukul. He extended a nice work upon persuasion of his PhD thereupon for last 10 years. And he learned a lot molecular and modern techniques located when he visited Switzerland also. So many congratulations to you.
Thank you.
Yeah.
Hello. Nice talk. Actually, I like these VOCs, the volatile compound. You work with that. So it was really nice. My question is whether this communication between the plants and the microbe, is it specific?
Specific in what sense?
The specific means a particular plant,
Yeah.
suppose in particular condition,
Definitely.
it is attracting a particular kind of microbe.
Definitely.
So what determines this specificity?
Like like I mentioned about the benzoxinoid, you can means
Like,
benzoxinoid.
let me give you one example.
Yeah.
So there is one microbe that is we use like agrobacterium, right? So whenever the plant is wounded, plant release acetylsyringol,
Right.
and that attract agrobacterium.
Right.
Similarly, is the same thing happen with the other microbes in the
Definitely.
microbiome, in
Yes.
the rhizosphere, especially?
Yes, sir. There are several reports about benzoxinoids that produced by the maize plant. And that benzoxinoids is attracted particular mycorrhiza that is particular specific for the maize plant. And they attracted different type of beneficial rhizobacteria also. And this is only because of the benzoxinoid. Otherwise, if we search the literature, benzoxinoid is a phytochemical that is mainly reported for the allelochemical properties, but still it attracts the bacteria. That is the interesting thing. Because if we talk about the allelochemical, that means they will kill all the microbes. But still this benzoxinoid attract mycorrhiza as well as some beneficial microbes also. So yes, there is a specific interaction between plant and microbes, because each plant have their own pattern of root exudates, and these root exudates attract individual microbes.
In this way, we can actually manipulate the microbiome.
Yeah.
Right.
Yeah.
And so that we can actually increase their efficiency toward the plant growth promotion.
Yeah, sure.
Right. If we know that it is attracting a particular kind of microbe in particular stress condition.
Yeah, that is people's working on synthetic microbial community.
Very nice work. Congratulations.
Thank you.
We can proceed further, Ayushi. So candid rendezvous and so forth, we play outside for a tea, and thereupon we shall meet tomorrow based on consensus to all of us. Yeah, one more thing, most important thing. Felicitation of my colleague, Dr. Anukul. Please call.
Thank you, sir, for an informative presentation. Now we would like to request Dr. Naveen Joshi and Dr. Swati Tripathi to felicitate Dr. Anukul Vaishnav.
With this, thanks to all my colleagues, students, scientific fraternity, IT peoples, admin staff, and so forth. We shall meet tomorrow at 10 a.m. See you till then. Thank you.